Are you okay? Okay. So, good evening, everyone. Good evening, everyone. आज से हम एक करंट अफेयर्स का एक नया बैच भी स्टार्ट कर रहे हैं. तो सम ऑफ यू मे बी अटेंडिंग. It's for some of you this may be the first class. I'm not going to introduce the class again. What are all the things that we're going to do? For those of you who are watching on YouTube, in the description of this video, there's an introduction video talking about all the components of the program. What are all the things that we're doing. Uh, for today's class, because the class is live on YouTube, in the description of this video, we have a Telegram channel link where the notes October part 1 and October part 2, both of them are there. You can download it from there. So whenever you start watching any of my class, please ensure that you have notes in front of you. I'll not dictate a lot of things. I'll rather focus on making you understand the concepts, making you understand the issue, rather than make you write things in the classes. So today we have two booklets, October part 1, October part 2. I would be starting with October part 2 because it's about Nobel Prize. Most of the topics there is about Nobel prizes given recently. So 5 Nobel Prize दे दिए गए हैं. 6th Nobel Prize, that is in economics. It's not actually a Nobel Prize, and we'll understand why it's not actually a Nobel Prize, and it will be given on 14th October. तो उसके अलावा all the 5 Nobel Prizes for this year have been given. We will be covering those Nobel Prizes in detail. Some of the things will be useful for preliminary examination. Nobel Prize क्या है, कहाँ मिलता है, क्यों मिलता है, for what areas which it is given, which are the bodies which are involved in giving those Nobel Prizes. But some things will be very important for mains examination also, like microRNA has got Nobel Prize this year. So microRNA का application in the field of medical science, that is very crucial. In the same way, the developers of artificial neural network have been given Nobel Prize. So artificial neural network, uske associated significance, that will be very crucial. In the same way, developers of AlphaFold 2 has been given Nobel Prize for chemistry. So AlphaFold 2 ने कैसे protein development में role play किया. These are all the things that we have to cover. Okay, but let us first start with छोटा सा, पहले briefly let's understand the all about Nobel Prize, all about the basics of Nobel Prize before we go into details of other things. पहले prelims facts में, in the end we are talking about the basics about Nobel Prize. We'll first do that and then we'll come to the science and technology portion. So देखिए, when it comes to intellectual achievement in the world, the most respected prize, most recognized prize, the highest prize is the Nobel Prize. It is given in the field of physics, chemistry, medicine or physiology, literature and peace. There is another prize जिसको हम Nobel Prize in economics बोल देते हैं, but that's not Nobel Prize as such. उसके बारे में बात करें तो economics में भी it's kind of given. Now, Nobel Prizes are given, or the Nobel Prizes were established by Alfred Nobel. Now, Alfred Nobel, what he did, towards the last phase of his life, in his will, he gave most of the things to Nobel Committee कि यार you take care of it and ensure that through this the most important contribution towards humankind is recognized and they are benefited from it. So his will in 1895 had provided for Nobel Prizes, and first Nobel Prizes were given in 1901. Later in 1968, Sweden का जो central bank है, it started another prize, Sverig's Riksbank Prize in Economic Sciences in memory of Alfred Nobel. तो देखो, rest of the 5 prizes were initiated by Alfred Nobel himself, whereas this was initiated by the Swedish Central Bank in recognition of Alfred Nobel. And thus इसको भी कई बार Nobel Prize in Economics बोल दिया जाता है. तो the first 5 prizes started in 1901, rest started in 1968. Now let us know a bit about Alfred Nobel. देखो ये prelims वाला, you have all got your notes, है ना? Towards the end of in prelims facts we are covering it. So Alfred Nobel was a Swedish scientist, chemist, entrepreneur. In fact, a number of things that we see today. आपने बोफोर्स गन के बारे में सुना होगा. बोफोर्स स्कैम हुआ था राजीव गांधी के टाइम पे, और बोफोर्स गन्स which were used in Kargil war. आप Kargil war पे when you watch any movie, वहां पे Bofors guns are used by India. Those guns were developed by those guns are developed by a company called Bofors. Alfred Nobel owned the company. Just imagine the impact that this person has had. Okay, so he was an entrepreneur, he was a chemist, he was a scientist, he has developed many things. He had more than 350 patents. His most important development or invention was dynamite. Actually, what he did, nitroglycerin was an unstable compound. He mixed nitroglycerin with other material to make it more stable so that it could be used for explosive purpose in a systematic manner. And this kind of made him very rich, because dynamite was much more useful than gunpowder. So dynamite started getting used in construction sector, it started getting used in mining sector, but most importantly in defense sector, which made him so rich that in the end he will give up, in his will he will say ki yaar, this money should be used to recognize those intellectuals who have contributed a lot towards humanity. Now, why Nobel Prize? तो इसके बारे में you would or why these prizes? Why Alfred Nobel decided to create these prizes? इसमें ना you will keep hearing about this legend that Alfred Nobel saw in a newspaper, like a newspaper has mistakenly published that Alfred Nobel was dead and it had criticized his contribution. तो जो legend है वो उसमें ये बोला जाता है कि his brother had died and some newspaper confused that with Alfred Nobel and they criticized him after his death. The merchant of death is gone or something like this kind of criticism was there, but there is no historical evidence for this. We keep on hearing this in the form of legends, but there is no historical evidence of this. There is no newspaper which someone can show कि देखो at the time of his brother's death it was by mistakenly assumed that Alfred Nobel was the person who had died and accordingly he faced all these challenges. Accordingly he had to go through this thing. So he created it. Perhaps he himself realized that his legacy should not be dynamite. His legacy should be something more important towards the humankind. Now, we'll look at a few things, but you'll see that of the 5 prizes, of the 5 Nobel, one though was created later, of the 5 prizes, 4 are given by Sweden. Sweden में ही अलग-अलग bodies हैं. We'll study about those bodies, we'll talk about those bodies, but 4 of those prizes are given by Sweden. Only for peace prize he chose Norway. आप देखोगे बाकी सब, you will see, for reasons that are not entirely clear, Nobel decided that the peace prize should be awarded in Norway and not in Sweden. Why was that done? Perhaps Sweden had a lot of military history or war ka history. So peace prize, he thought that if it's given by Norway, it would be more, it would be making more sense. Now, who awards these prizes? ये preliminary examination के लिए you should know. देखो, Alfred Nobel created 5 of these awards, but eventually which are the bodies which are responsible for awarding it, recognizing who should be getting it, and choosing who should be getting it and awarding it. So there is one, Royal Swedish Academy of Science. In fact, he was also a member of it. Alfred Nobel was one of the members of it. This Royal Swedish Academy has been made responsible for physics and chemistry prizes. By Alfred Nobel, in his will, it was provided that physics and chemistry should be given, physics and chemistry awards should be given by him. Then, when this bank created which prize? Economy prize, uska responsibility was also given to Royal Swedish Academy of Science. Got it? Pehla aspect. Then, who gives medical wala prize? Nobel Prize in Medicine. So, Nobel Prize in Medicine was is given by 50 professor at the Karolinska Institute, which is called Nobel Assembly. ये मेडिसिन भी कहाँ पे? स्वीडन में दिया जाएगा. So this institution, Karolinska Institute, will also be in Sweden. Then literature is given by Swedish Academy. But when it comes to Nobel Peace Prize, Norwegian Nobel committee is responsible for it. एक लाइन में पता है, like last year a lot of factual things were asked by UPSC. So I do not want कि there should be something जो वो पूछ ले और हमने कभी सुना भी ना हो, कभी देखा भी ना हो. तो इवन इफ यू रिमेंबर और रिकॉल दिस, यार पीस प्राइज तो नॉर्वे से आ रहा होगा, यू विल बी एबल टू एलिमिनेट सम थिंग्स, यू विल बी एबल टू एलिमिनेट सम एस्पेक्ट्स. वो मेरा पर्पस है, बाय इंश्योरिंग दैट यू आर ब्रॉडली गोइंग थ्रू इट. नाउ इस साल प्राइस किसको मिला है? देखो, दीज 3 प्राइजेस वी आर गोइंग टू स्टडी इन डिटेल साइंस एंड टेक्नोलॉजी में. कौन कौन सा? मेडिसिन वाला, फिजिक्स वाला एंड केमिस्ट्री वाला. मेडिसिन प्राइज हैज बीन गिवन फॉर discoverers of micro RNA. So we'll today understand what micro RNA is, what its purpose is, what its significance is. It has been given to Victor Ambrose and Gary Rufkund. The Nobel Prize in Physics, this year this has become a bit controversial. Nobel Prize in Physics is a bit controversial this year because it has not been given to physicists. It has been given to computer scientists. जैसे John H. Hopfield, he was the first person who kind of created a preliminary type of artificial neural network. We'll understand that. Whereas this person, Geoffrey E. Hinton, he is considered the godfather of artificial intelligence. In fact, till 2023, he worked at Google in the field of artificial intelligence. In 2023, he quit Google because he wanted to openly criticize artificial intelligence. और उस टाइम भी इनके बारे में बहुत सारे, बिकॉज द गॉडफादर ऑफ आर्टिफिशियल इंटेलिजेंस हिमसेल्फ इज क्रिटिसाइजिंग इट. So we will understand this also in detail. And then Nobel Prize in Chemistry was given to David Baker and Demis Hassabis, John M. Jumper. It's about proteins. So अभी हम देखो, this person was able to create a new protein altogether, and these two people again come from the field of artificial intelligence. इस बार देखो chemistry में भी artificial intelligence का role है. AlphaFold 2 is an artificial intelligence tool developed by a company called DeepMind. This company is owned by Google, and they have developed this tool to predict the structure of protein. अभी like you may not be able to exactly understand the significance of that. अभी इनको detail में पढ़ेंगे. So the developers of AlphaFold 2 have also been given Nobel Prize here. But Nobel Prize in Chemistry, protein manufacturing और protein का structure study कर पाना, that's part of chemistry. Okay, then Nobel Prize in Literature किसे मिला? Nobel Prize in Literature this year has been given to Han Kang. She's a South Korean. She's the first person from South Korea to receive a novel in literature. She's also the first Asian women to be given Nobel Prize in Literature. इतना हमें पता होना चाहिए. First Asian women. Asian men तो हैं जिन्हें लिटरेचर में नोबेल प्राइज मिला है. फ्रॉम इंडिया हु इज इट? रविंद्रनाथ टैगोर, है ना. लिटरेचर में उनको नोबेल प्राइज मिला है. बट एशियन women to get a Nobel Prize for the first time is this woman. Okay, then एक और चीज जो नोबेल पीस प्राइज है उसके बारे में भी थोड़ा सा देख लेते हैं. नोबेल पीस प्राइज हैज बीन गिवन टू अ इंस्टीट्यूशन कॉल्ड निहॉन हिंडांक्यो. It has been given to this institution called Nihon Hindankyo. What is this institution? देखो, इस institution का आप इंग्लिश में नाम देखोगे ना, the Japan Confederation of A and H Bomb Sufferers Organization. इसका जापानीज में द नाम है Nihon Hindankyo. So these are basically this organization was set up in 1956 with two objectives in mind. जापान में हिरोशिमा नागासाकी पे न्यूक्लियर बॉम्ब्स गिरे थे, तो there were survivors also. A lot of people died, but there were survivors also. उनका तो one purpose of this body was to promote the social and economic right of all hibakusha. Hibakusha are survivors of Hiroshima and Nagasaki, and to ensure no one ever again is subjected to catastrophe that befell the hibakusha. The issues that they faced, that is nuclear attack that they face, no one should ever be facing it. And for this, what they are doing, they are through personal witness statements spreading awareness against use of nuclear weapon, and thus are trying to promote peace in the world. For that purpose, they have been given Nobel Prize in Peace this year. Okay, तो prelims के लिए ये चीजें हमें याद रखनी है. On 14th, that is on Monday, you will have Nobel Prize for Economics. But हम अब पढ़ते हैं Nobel Prize कहाँ पे? Nobel Prize from the perspective of science and technology. Nobel Prize in Medicine, what has it been given for? What is its significance? Nobel Prize in Physics, Nobel Prize in Chemistry, what are all the things for which it has been given for, and how will those things be useful for us? है ना? पहले शुरू में कुछ factual चीजें before we actually jumped into detail of science and technology, I wanted to cover. Broadly, ये पढ़ना है, इसे ignore नहीं करना है, because when कुछ भी random आ जाएगा examination hall में, you should have at least read it once, so that when it comes in front of you, you'll be like, हाँ, मैंने ये टर्म सुना है. When even if you are, let's say, not revising it just before examination, but when you have heard Nihon Hindankyo, ये term, when it comes in the examination, there are chances that you will be marking it correct. आप सोचोगे आपने तुक्का मारा है, but वो तुक्का नहीं होगा. It will be based on your knowledge that or your static knowledge that you have, जो आप उस time immediately recall नहीं कर पा रहे हो, but you'll be able to do better. Now, let's start with the first topic. Nobel Prize in Medicine. Nobel Prize in Medicine. यहाँ पे मैंने एक subjective question भी दिया है. What is microRNA? Discuss its significance for human physiology and medical science. इस तरह का question मेंस एग्जामिनेशन में आता है. One thing that we should be ready for is this, and then preliminary examination के लिए भी सारे aspects से we should be ready for. Shashikant Das is asking, hello sir, will you cover CA of previous month? Yes. देखो, in this program, it's my responsibility to ensure that everything that you need for your preparation for prelims before preliminary examination and for mains before mains examination would be done. You do not have to worry about this because a batch is starting in October, will not be doing something which happened in May, June, July, August, or will not do something which is 2 years old. कोई चीज 2 साल पुरानी भी है, but आज इस साल के एग्जाम के लिए relevant होगी, मैं आपको वो करवा के दूंगा. Please understand this. The USP of this program is current affairs is my responsibility. You do not have to do anything. मैं next class में मैं योजना और कुरुक्षेत्र करने का प्लान कर रहा हूँ. इस बार योजना स्वच्छ भारत अभियान के 10 years पे आया है. कुरुक्षेत्र हेल्थ एंड न्यूट्रिशन पे आया, तो वो भी करवा के देने की मेरी जिम्मेदारी है. पुराने वालों में भी जो रेलेवेंट है वो करवा के देने की मेरी जिम्मेदारी है. इट्स योर करंट अफेयर्स इज माय रिस्पॉन्सिबिलिटी. एवरी संडे यू हैव 2 टेस्ट. आपको कल शाम को सिलेबस मिल जाएगा. एवरी संडे यू हैव 2 टेस्ट्स. इंश्योर that you are preparing for those tests systematically. Will also ensure that you are revising everything अच्छे से. You'll have weekly test, you'll have monthly test, you'll have 3 monthly test. In this way, आप weekly test में पहली बार रिवाइज करोगे, monthly test में दूसरी बार रिवाइज करोगे, 3 monthly test में तीसरी बार रिवाइज करोगे. You'll be able to revise everything in a very timely manner, and those tests will also have static portion, keeping you in sync. Okay. Will this class be continue on YouTube? नहीं, it won't continue on YouTube. आज की और कल की क्लास we are keeping as demo on YouTube. If I go on YouTube, then why would students pay fee and make it happen? चलो छोड़ते हैं अभी. Let's come to this Nobel Prize in Medicine. किसे मिला है? Nobel Prize in Medicine has gone to Victor Ambros and Gary Ruvkun for the discovery of microRNA. What is microRNA? What is the purpose of microRNA? अच्छा, before we understand microRNA, क्योंकि microRNA में protein synthesis के बारे में थोड़ी सी बात होगी. Do we understand how protein is formed in our body? Do we all understand how protein is formed in our body? देखो, we have cells. Within the cell, we have nucleus. Nucleus में we have DNA. Some DNA we got from our father, some DNA we got from our mother. Some chromosomes we got from our father, some chromosomes we got from our mother, and उसने हमें एक unique DNA दिया है. And that unique, do I need to do cover vision PT 365 prelims? नहीं, you do not have to. मैंने बोला ना, it is my responsibility to ensure that everything that you need for this exam. देखो, कोई बच्चा जो हमसे पास्ट में पढ़ा है ना, उससे पूछना. They will tell you, they will be the right guide rather than मैं बोल रहा हूँ. They will tell you this कि इतना पढ़ के चले जाओगे वो बहुत होगा. It's my responsibility to go through all the material and to ensure that कोई भी चीज जिसमें relevant है वो आपके नोट्स में रहेगी. आपको मैं ना एक वीडियो बना नहीं पाया हूँ, मोस्ट प्रोबेबली मैं कल बनाऊंगा. इनफैक्ट कल अगर समय रहा तो मैं लाइव क्लास में ही करता हूँ. ये वो वीडियो था इस बार मेंस एग्जामिनेशन में कितने क्वेश्चंस करंट अफेयर्स प्रोग्राम से है. अब सब बोल देते हैं देखो मैंने ये टर्म पढ़ाया था, ये पढ़ाए थे. वैसे नहीं, एग्जैक्टली जो पढ़ाया है उससे कितने क्वेश्चंस आए? वी हैड 17 क्वेश्चंस फ्रॉम करंट अफेयर्स प्रोग्राम. स्टूडेंट्स वर वरीड अबाउट कि टारगेट ऑफ सेंडाई फ्रेमवर्क. हमने टारगेट ऑफ सेंडाई, सातों टारगेट एकदम बच्चों को रटवाए थे क्लास में. क्यों रटवाए थे? क्योंकि सेंडाई फ्रेमवर्क पे सवाल आने का चांसेस ऑल हमेशा रहा है और बहुत इजी था. इस बार करंट अफेयर्स में किसी ने उड़ान स्कीम नहीं पढ़ा हुआ था, हमारे बैच में पढ़ा हुआ था. इस बार मेंस एग्जामिनेशन में उड़ान स्कीम पे क्वेश्चन आया है. माय पॉइंट इज कि we try to ensure that each and everything which is relevant for exam would be taught to you, and किसी और सोर्स को पढ़ने की जरूरत नहीं है. we'll make sure that everything is there in your notes. ensure, मुझे एक प्रॉमिस चाहिए कि आपको जो पढ़ाया गया वो आता है. I can promise you that will be enough for your selection. अगर आप प्रिलिमिनरी एग्जामिनेशन की बात करते हैं, अगर आप प्रिलिमिनरी एग्जामिनेशन में जो आपको टेस्ट में असाइनमेंट मिल रहा है, स्टैटिक पोर्शन आपको खुद से पढ़ना होगा. हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी का बेसिक्स वो आपको असाइनमेंट के फॉर्म में मिलेगा. अगले संडे के टेस्ट में यू विल गेट हिस्ट्री एंड पॉलिटी एंड इकोनॉमी पोर्शन ऑल्सो, स्टैटिक वाला जो आपको खुद से पढ़ना है. एंड विल हैव करंट अफेयर्स पोर्शन ऑल्सो. In this way, we'll go ahead to if you keep revising, cover the static portion basic and follow the current affairs properly, you will have enough marks in your preliminary examination, enough marks in your mains examination. Will there be discount for those who have once registered for any of the course last year? देखो, अगर जो करंट अफेयर्स वाले बच्चे हैं लास्ट ईयर वाले, उनके लिए तो कुछ डिस्काउंट है, बट उसके लिए आपको ऑफिस में कॉल करना पड़ेगा. लेट्स कम टू प्रोटीन वाला बात. हम प्रोटीन फॉर्मेशन की बात करते हैं. देखो, हाउ इज प्रोटीन फॉर्म्ड इन आवर बॉडी? तो वी हैव सेल के अंदर न्यूक्लियस है. नाउ लेट मी मेक अ बिगर सेल. लेट मी से this is the cell and this is the nucleus. Inside the nucleus we have DNA, and on DNA we have genes. Those genes are basically encoding for protein. Those genes are basically encoding for protein. They have coded for a protein. So which protein will be formed in my body will be decided by what gene I have. But let me draw this DNA. DNA के ऊपर gene होता है. We have 25,000 genes, and thus we can have 25,000 different protein. Now, protein is manufactured outside the cell in protein factory called ribosomes. Protein is manufactured outside the cell by protein factory called ribosomes. They are not manufactured inside the cell. Where is the information given of which protein has to be formed? Inside the cell. Genes don't come out. तो यहाँ पे क्या होता है? Genes से एक messenger RNA बनता है. And this messenger RNA goes out. It goes out and tells ribosomes this protein has to be formed. Okay, अभी we will when we study biotechnology from basics. जनवरी में हम साइंस एंड टेक्नोलॉजी पूरा पढ़ना शुरू करेंगे, एकदम स्टैटिक सब कुछ पढ़ते हुए. लाइक फॉर सम सब्जेक्ट्स मैंने आपको बोला है आपको हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी खुद से बेसिक्स उसका खुद से पढ़ना है. हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी का मैं आपको करंट अफेयर्स पढ़ाऊंगा खाली इन करंट अफेयर्स प्रोग्राम. बट फॉर सब्जेक्ट्स लाइक साइंस, एनवायरमेंट, इंटरनेशनल रिलेशंस, इंटरनेशनल बॉडीज, मैपिंग, आपको कुछ नहीं पढ़ना है. इसका मैं सब कुछ पढ़ाऊंगा आपको, इवन द बेसिक पोर्शन भी पढ़ाएंगे. ओके, तो लेट्स कम बैक. हाउ इज प्रोटीन फॉर्म्ड? हमारे सेल के अंदर न्यूक्लियस है. न्यूक्लियस में डीएनए है which we have got from our parents, and on those DNA, parts of DNA, sub parts of DNA are known as genes. Those genes encode for protein. Those genes have code for protein. Protein कैसे बनता है? Protein is basically a chain of amino acids. तो amino acid कौन कौन से जोड़ के, which are the amino acids which have to be brought together to form a protein in your body? Protein which is formed in my body is different from protein formed in your body. देखो, मेरा स्किन कलर आपके स्किन कलर, मेरा हेयर कलर आपके हेयर कलर, हम सबका यूनिक है because different kind of protein is being formed in our body. Okay, so my question was कि how is protein formed? Protein is formed with the help of messenger RNA. तो जीन से, डीएनए से जब ये आरएनए बना है, इस प्रोसेस को हम बोलते हैं ट्रांसक्रिप्शन. फिर आरएनए से जाके प्रोटीन बनेगा. इतना तो समझ में आया हमें? Are we all clear about it? Okay. Now I have another question. Question is, do all our cells in our body there are billions of cells. Do all our cells have the same DNA? except let's say red blood cells जिसमें न्यूक्लियस होता ही नहीं है, बट जनरली क्या होगा, हमारे सारे सेल्स में सेम डीएनए है. व्हाई? बिकॉज व्हेन यू थिंक ऑफ हाउ आवर लाइफ बिगैन, इट बिगैन एज अ सिंगल सेल. मदर्स एग फर्टिलाइज्ड बाय फादर स्पर्म, वो अपने क्रोमोसोम्स लेके आए, ये अपने क्रोमोसोम, स्पर्म में क्रोमोसोम्स आए, फर्टिलाइजेशन हुआ, एक यूनिक सेल बन गया, फर्टिलाइज सेल, जिसे हम जाइगोट बोलते हैं. आफ्टर दिस व्हाट हैपेंस? इट इज दिस सेल ओनली व्हिच कीप्स ऑन मल्टीप्लाइंग. So it will all have the same nucleus and thus the same DNA, right? So every cell in our body has the same DNA. Within the nucleus has the same DNA. Are we clear about this? It has the same genes. Are we clear about this? Fine. But do every cell in our body produce the same protein? No. Think of this, the cells which are here, आपके hair follicles में, वो तो ये बाल बना रहे हैं, but the cells which are in your heart, are they creating these hairs, protein for these hairs? नहीं ना. अगर वैसा होता तो जब दिल निकाला जाता बाल बाल वाला दिल आता, right? Every cell is not making the same protein. Are we clear about it? Every cell is not. Our pancreas are making different protein, our heart is making different protein, our heart cells are making different protein, our skin cells are making different protein, our follicles are making different protein, right? So not every, but उन सब में DNA same है. Take out any cell from your body and you will find exactly the same DNA which was there when the fertilization had taken place. जब ये single cell, ये egg cell, all the life on earth starts with a single cell. Even all of us, now today we have billions of cells in our body, but we began as a single cell. Mother's egg cell was fertilized by father's sperm to form a zygote. This is how we started, and this zygote only has multiplied to create this complex human being. So all the cells in our body have the same DNA. So let's say this is the DNA which is there in all the cells of our body. Now parts of DNA are known as gene and they are encoding for protein, but not all the proteins are getting expressed in every cell. इतना तो समझ में आ गया? Not all the proteins are expressed in every cell. Our pancreas में जो cells है, they are expressing some other protein. So they have gene 1, they have gene 2. ऐसे ही they may have 20,000 different genes, 20,000 plus different genes, but not all genes are getting expressed. Not all genes are getting expressed in every cell. In different cells, different genes are getting. हमारे in our body are all cell types the same? No. Our muscle cells are different, our heart cells are different, our pancreas are different. Pancreas में जो cells are different है, our neuron cells are different, our skin cells are different. सारे cells भी same नहीं है. The protein which they are manufacturing is also not same. What is same? Their DNA. उनके न्यूक्लियस के अंदर जो DNA है, that is same, but they are functioning differently. How this regulation happens is the subject of this year's Nobel Prize in Medicine. Do we get it? Do we understand it? So if you'll come here, basic question was, our organs and tissues consist of many different cell types, all with identical genetic information stored in their DNA. However, these different cells express unique set of proteins. They are not expressing the same set of protein. देखो, different cells हमारे brain में जो cells हैं, cell type, it is creating, expressing some other protein. Muscle cells are expressing some other proteins. So cells are same, genetic material is same, but the proteins which are being created by them are unique, are different. Not all proteins are different. Some proteins may be same, but different types of cells are also creating different kinds of proteins. How does that happen? Okay, this is the problem that the current scientist solved. पहले problem समझना बहुत जरूरी है. Only then we'll be able to understand how they have solved it, and only then we'll be able to understand what is the significance. पहले the things that we are studying right now, the things that we are trying to understand right now, that will give us very fundamental basic understanding, so that no matter what question gets asked in the examination, we'll be able to solve it easily for preliminary examination. But आखिरी में हम पढ़ेंगे 5-6 significance of this that will be relevant for our mains examination. इसका application क्या है? Because mains examination में questions कैसे आते हैं? Application, significance वाले. Whereas preliminary examination में they'll ask you anything. RNA interference technology से इनमें से क्या हो जाएगा? या micro RNA contributes in which of the following? और तो you have to know micro RNA from fundamentals to actually able to recognize ये नहीं हो सकता, ये हो सकता है, because they may give you anything, whereas there they'll ask you give me applications, and you will be able to write your own applications of micro RNA in the mains examination. Okay, we understand the problem. Now, before discovery of micro RNA, when scientists were asked कि यार ऐसा क्यों हो रहा है, like how is it happening that some cells are producing some kind of protein, other cells are producing other kind of protein. How does that work? So, in 1960s, the concept of transcription factors was used. It was said that it is this transcription factor which are responsible for some cell expressing some protein, other cell expressing other protein. So, what are these transcription factors? These are specialized proteins found in our body, and they bind to specific region in DNA. So basically यहाँ पे image नहीं है. Let me come here. This is my DNA. We're saying this transcription factors are basically proteins which will come and bind on our DNA. By this, they will control whether this gene will be expressed more or this gene will be expressed less. So what are transcription factors? पहली चीज, if you are asked what is transcription factor, transcription factors are protein which controls whether a gene will be expressed more or a gene will be expressed less. In our heart cells, बाल बनाने वाले प्रोटीन का gene will not be expressed. तो वहां पे transcription factor, it can bind to specific region in DNA. So our transcription factor binds to the specific region in DNA and control the flow of genetic information by determining which mRNA are produced. Gene से क्या बनता है? mRNA बनता है, so it will control which mRNA is getting produced, and thus is controlling which protein is getting produced. Because जिस RNA का, जिस DNA का mRNA बनेगा, mRNA only is taking information to the ribosomes to make this protein, manufacture this protein. तो जिस gene का mRNA बनेगा, वो ही only that gene is going to get expressed. So this factor, transcription factor, controls which mRNA will be formed. Okay, now transcription factors can be of two types. One is activator, other is repressor. Let's say this is my DNA and इस gene का transcription factor आके यहाँ पे attach हो रहा है. If this transcription factor is activator, it will lead to more formation of mRNA, and thus this gene getting expressed more. अगर ये transcription factor activator है, it will boost the production of mRNA and thus boost the production of protein. Whereas if this transcription factor is a repressor, it will resist or decrease production of mRNA and thus decrease the production of protein. ये बात समझ में आई? Did we understand the meaning of transcription factor? Are we all comfortable with the term transcription factor? So what is transcription factor? Transcription factors are proteins which get attached to DNA and restrict or promote formation of mRNA. वो जिस जीन के डीएनए के जिस कंपोनेंट पे अटैच होंगे उसमें दे कैन वर्क एज एक्टिवेटर और दे कैन वर्क एज रिप्रेसर. तो in 1960s when it was understood कि why a cell is forming this protein, manufacturing this protein, whereas the other cell is manufacturing other protein, our understanding was on the basis of transcription factors. It is transcription factors which are controlling everything, because at that time we did not know about microRNA. Okay. However, in 1993, this year's Nobel laureate, this year means 2024 के जो Nobel laureates हैं, they published unexpected findings describing a new level of gene regulation. Which gene is getting expressed, which gene is not getting expressed is gene regulation, and they, by discovering microRNA, explained other mechanisms by which gene is getting regulated. Okay, how did these scientists contribute? Let us understand. अभी तक हमने background ही समझा है. Before microRNA was discovered, what was considered the thing which controlled which gene is getting expressed, which gene is not getting expressed? Transcription factors. So, do transcription factor really exist? So, yes, they exist. Do they regulate gene expression? Yes, they regulate gene expression. They also function, but what also functions? MicroRNA. In 1993, they, for the first time in a research paper, published this year's Nobel laureate published about microRNA. Basically, they were working on C. elegans. It's a roundworm, a very small roundworm. They were working on it, but this roundworm, though very small, produces various kinds of cells. इसके पास muscle cells हैं, neuron cells हैं. So through the study of this development of this roundworm, they were trying to understand when does a cell, we were initially what? A single egg cell. It has specialized into muscle cell, neuron cell by producing different proteins. So how does this differentiation occur, and what is responsible for formation of this protein? Before this, what was the reason given? Transcription factor is the reason. But the two scientists were interested in genes that control the timing of activation of different genetic program. That is, when different genetic programs start getting expressed in our body. Initially, egg cell mein same DNA, same protein, but it specializes into different kinds of cells as multiplication happens. So how that is happening, they were trying to understand that. And it was Victor Ambrose who in his lab at Harvard discovered microRNA, a short RNA molecule that lagged code for protein production. This microRNA will do what? Come here. He discovered microRNA. This microRNA will not prevent the formation of mRNA. Transcription factor was doing what? Transcription factor was preventing or promoting the formation of mRNA. It did not do that. Ye microRNA he found after mRNA has been formed, it blocks the functioning of mRNA, and by that blocks some gene from getting expressed. So in my heart, the gene related to protein which will create hairs are not getting expressed. They are not getting expressed. Why? Because some microRNA is blocking the mRNA of this mRNA of the protein which creates my hair. Are we getting it? So he found this for what? He found this for roundworm. In his lab at Harvard discovered microRNA, a short RNA molecule that lagged code for protein production. So this RNA molecule itself did not produce protein, but Gary Rivkin found that microRNA did not inhibit the production of mRNA. MicroRNA is not inhibiting the production of mRNA. mRNA is getting formed, but the regulation appeared to occur at a later stage in the process of gene expression through the shutdown of protein production. So the microRNA is not preventing the formation of mRNA. The regulation is happening after that, after mRNA has been formed. That is what the other scientist Gary Rivkin found. Then two scientists came together, collaborated. Initially also they were collaborating in their postdoctorate period in 1980s. Then they moved to, after their postdoctorate, they moved to different places, but they were doing research in the same field. They collaborated and found that microRNA produced blocks that target gene by binding to complementary sequence in the gene's mRNA, which eventually blocks the production of protein. So basically, microRNA blocks the attaches to mRNA, which blocks the production of protein. It is what they found. Thus, they developed or they discovered a new method of gene regulation. अभी तक gene regulation का method क्या था? Transcription factor. Here, they new principle of gene regulation was found, mediated by previously unknown type of RNA, microRNA. Previously, microRNA were also not known. Here, they were able to find a new kind of RNA called microRNA. When it was published in 1993, scientific community was not very excited. 1993 में when microRNA was published for the first time, scientific community was not very excited because they thought that this was something which was a peculiarity of this roundworm. कि उस राउंडवर्म में रेगुलेशन ऐसे होता है, not in other complex organisms, so it's not going to affect humans. Are we getting it? Are we understanding it? So in 1993, when microRNA was discovered and it was published, scientists were not very excited because they said their study is on roundworms, and thus the microRNA system of regulation of genes happens where? In roundworm. But the perception changed in 2000 when Ruvkun's research group published their discovery of another microRNA which is found in even complex organisms including humans. And since then, more than 1,000 genes for different microRNAs in humans have been found. So, hamare, like in human body only, there are around 1,000 microRNAs which have already been found. Please understand, microRNA will be formed from what? From DNA? Please, समझो इस बात को. What we have DNA. From DNA, mRNA is formed. From DNA only, microRNA would be formed. So, since 2000, scientists in India, also India बोल रहा हूँ, scientists studying human beings have found more than 1,000 genes on our DNA, जिससे क्या बनता है? microRNA, and thus more than 1,000 microRNAs have been recognized even in humans, and this becomes exciting. In 1993, when we said कि वो roundworm का peculiarity है, what? microRNA is a peculiarity of roundworm. It will regulate gene expression in roundworm. Then no one was interested. What will we do? But when we realize, okay, this is impacting gene regulation in not only roundworms but also in other complex organisms including human beings, it becomes very significant for us. It becomes very important for us. Now, further research in this field, so we understood कि perception तो change हो गया, this is going to be significant. So further research in this field, you'll see in addition to mapping of new microRNA, experiments by several research groups elucidated the mechanism of how microRNAs are produced and delivered to complementary target sequence in regulation of regulated mRNA. Basically, they're saying is they discovered mRNA. After that, research happened to understand how microRNA are actually produced and how those microRNAs are delivered to the complementary messenger RNA, because microRNA are going to do what? They're going to block messenger RNA, so they have to be delivered. So scientists in future also found that. Now, something very important, इसमें ना prelims में question बनते हैं, या मैं तो एटलीस्ट बनाने का प्लान कर रहा. One is a single microRNA can regulate the expression of many different genes. एक single microRNA can regulate many different genes. A single microRNA is not for only one gene. It can regulate many different genes. And a single gene can also be regulated by multiple microRNAs. Are we understanding this? What I'm trying to say is कि this was gene 1, this was gene 2. Let's say this was a gene for microRNA. इससे बनता है expression से, transcription से microRNA. This microRNA 1, and let's say there is another microRNA 2. This microRNA 1 may be regulating gene 1 also, may be regulating gene 2 also, gene 7 also, gene 10 also. Right? One microRNA may regulate multiple genes. How? By restricting the function of their messenger RNA. इस जीन का मैसेंजर आरएनए बनेगा उसके फंक्शन को रेगुलेट करके. तो वन माइक्रो आरएनए इज नॉट स्पेसिफिकली फॉर वन मैसेंजर आरएनए. In the same way, one gene or one messenger RNA can be blocked by multiple micro RNAs. It's not that only microRNA 1 will be blocking messenger RNA 1. Messenger RNA can also be blocked by multiple of them. इससे भी ब्लॉक हो सकता है, इससे भी ब्लॉक हो सकता है. Do we get it? Yes. Every नहीं एवरी वही तो बताया ना, एवरी जीन के पर्सपेक्टिव से कोई इंडिविजुअल माइक्रो आरएनए होने की जरूरत नहीं है. वन माइक्रो आरएनए में वर्क फॉर मल्टीपल जीन्स. ओके. यस. नहीं, वन जीन कैन बी कंट्रोल्ड बाय मल्टीपल आरएनए. सिर्फ आरएनए माइक्रो आरएनए 1 से ही कंट्रोल नहीं होगा. ये माइक्रो आरएनए 2 से भी हो सकता है, माइक्रो आरएनए 5 से भी हो सकता है, माइक्रो आरएनए 10 से भी हो सकता है. व्हाट इज द great? What I'm trying to say is, let's say this is gene 1, this is gene 2, this is gene 3. ऐसे ही we have gene 20,000. इतने जीन्स हैं हमारी बॉडी में फॉर मेकिंग प्रोटीन. वी हैव माइक्रो आरएनएस, है ना. माइक्रो आरएनए 1 से लेट्स से माइक्रो आरएनए 1000. वी हैव मोर देन 1000 माइक्रो आरएनए, वी हैव मोर देन 20,000 जीन्स, है ना. एंड फॉर एवरी जीन वी हैव व्हाट? मैसेंजर आरएनए. यहाँ पे माइक्रो लिखना चाहिए, एम नहीं. एम रेफर्स टू माइक्रो आरएनए 1 to माइक्रो, है ना. वी हैव मैसेंजर आरएनए, वी हैव मैसेंजर आरएनए. दिस मैसेंजर आरएनए, विल दिस मैसेंजर आरएनए बी यूनिक? हर जीन अलग प्रोटीन को एक्सप्रेस कर रहा है तो दिस विल बी यूनिक. इट हैज मैसेंजर आरएनए 3, 1. What I'm saying is, if you have to block messenger RNA 1, it may be blocked by more than one micro RNA. Micro RNA 1 में also block it, 5 में also block it, 10 में also block it. ऐसा हो सकता है. नहीं, एक ही, not together, है ना. Any one of them, not any one of them. इनमें से कुछ होंगे जो 1 को block कर सकते हैं. Not all of them will be able to block 1, but more than one may be able to block 1. In the same way, what we are saying is, when it comes to mRNA 2, it can be blocked by multiple of them. So that is what, ये क्या हुआ? It should be noted that a single microRNA can regulate expression of many different genes, and a single gene can be blocked by, so this one mRNA may block this also, may block this also, may block this also. In the same way, mRNA 2 is getting blocked by mRNA 1. It may also be getting blocked by this and this. Now do we understand? Do we understand this? Okay. So this is, इस पे ना, when I was reading this, when I was studying this, I thought एक अच्छा क्वेश्चन बनेगा. एक जब मैंने have to make MCQ में already लीक कर रहा हूँ. कोई बात नहीं, रिवाइज करो, मैं थोड़ा सा और मॉडिफाई करके I'll make an MCQ for next test. So now we have understood this. Now see, microRNAs used to block mRNA naturally. This process has been used to block messenger RNA in artificial mechanism also. This is known as RNA interference technology. हम किसी जीन का एक्सप्रेशन अपने आप से, like human beings artificially wants to do that. So we would want to create some compound which can block the expression of mRNA. Suppose one gene in our body is creating cancer, is leading to cancer. So we would want that उस जीन का एक्सप्रेशन बंद कर दिया जाए. That gene is creating messenger RNA if we block this messenger RNA, will this gene's expression be blocked? So this is one of the things that we do in RNA interference technology. In 2018 या 19 there was a question in preliminary examination on RNA interference technology. उसमें हम क्या करते हैं, मैसेंजर आरएनए को इंटरफेयर करते हैं सो दैट एक्सप्रेशन नहीं हो. तो मेथड टू डेवलप मैसेंजर आरएनए इंटरफेरेंस कैन बी लर्न फ्रॉम माइक्रो आरएनए, बिकॉज व्हाट इज माइक्रो आरएनए डूइंग? माइक्रो आरएनए इज इंटरफेयरिंग विथ messenger RNA's expression. So cellular machinery for producing functional micro RNA is also employed to produce other small RNA molecules in both plants and animals. For example, as a means of protecting plants against virus infection, Andrew J. Fire and Craig C. Mello were awarded Nobel Prize in 2006 for describing RNA interference. तो पता है RNA पे 3 बार नोबेल प्राइज मिल गए. RNA इंटरफेरेंस पे इनको मिला, अभी माइक्रो आरएनए पे अभी वाले लोगों को मिला, एंड लास्ट ईयर आरएनए वैक्सीन. in last 20 years only in medicine, RNAs have been able to, or the work on RNA have been able to get 3 Nobel Prize in medicine. Now we'll come to significance. अभी तक जो हमने समझा ना, वो all the things that we studied, we understood, वो हमें basic understanding में help कर रहा है. But now what we are going to study here will be something that we would directly want to write in our mains answer. Why this discovery of microRNA is significant, why this discovery of microRNA is so important, so relevant. One, it helps us better understand how our body functions, how gene get regulated. Earlier it was only transcription factor, but now this is also there. Then it has been at work for, it has also contributed in our evolution. How has it contributed in our evolution? See, initially life would have began as a single cell, है ना? फिर an organism which have same kind of cell, but then an organism which have multiple kinds of cells. Multiple kinds of cells means in those cells different genes are getting expressed. For that, what is needed? MicroRNA would be needed, because you want to block some gene, you want to allow some gene to get expressed. So microRNA has also played an important role in evolution, and thus studying microRNA can help us understand evolution also. Then genetic research show that cells and tissues don't develop normally without microRNAs. That is, if microRNAs are not there, or if microRNAs have some issues, then your body will not be able to develop properly, or abnormal regulation by microRNA. If your microRNAs are not functioning properly, you may suffer from cancer, cardiovascular diseases, diabetes, viral infections, and other such issues, other such challenges. And therefore, when it comes to treating these diseases, can microRNA play an important role? Can microRNA based therapies, because some microRNA is not functioning properly, so some microRNA based therapies can help solve those problems. It can also be excellent biomarker. What is biomarker? Biomarker is, it is telling if you are suffering from some disease, say if you are suffering from dengue, you will see platelet counts reduce हो जाते हैं. That's a kind of biomarker. ऐसे ही when you are suffering from certain microRNA में deficiency की वजह से challenges, some cancer which because your microRNA is not blocking some MR, then it can clearly tell you that this microRNA is creating problems. So this presence of this microRNA or absence of this microRNA can act as biomarker, can tell you what is the issue, what is the problem that you are facing. So microRNA can also act as excellent biomarkers. For example, in cancers, the level of certain microRNA are abnormal. These create unique signature pattern that allow identification of which kind of cancer are you suffering from. So it acts as biomarker also. Do we get it? MicroRNA के advantages, significance. At the same time, what do we need to do? We need to prioritize development of microRNA based therapies. Already a lot of research on this is going on. ऐसा नहीं है नोबेल प्राइज मिलने के बाद ही सबको पता लगा. Scientific community knew about microRNA and its significance in the past also, and a lot of research on this has been going on. microRNA based therapies, research is going on. We need to prioritize that because a lot of diseases can be solved by microRNA based therapies. Second, we should emphasize on developing suitable microRNA delivery system. Eventually, if some microRNA is not functioning properly, we'll have to deliver it within the cell, within the nucleus. उसके लिए we can use nanoparticles, we can use viral vector mechanism, we can use some other polymer for that. That thing needs to be developed. India should carry out studies on microRNA biology with the aim of producing clinically relevant molecules and diagnostics, and India should also enhance international collaboration. जैसे अभी we are collaborating on cancer moonshoot with USA. So we need to collaborate in the field of development of microRNA to get more technologies to solve more health related challenges of India in future, जिससे हम नई therapies, नए diagnostic, नए treatment के mechanisms develop कर पाएंगे. Okay, these are certain other things that we need to do. तो in conclusion, ये एक line मैंने शायद एक ORF में article था इस about mRNA, जो Nobel Prize है उसके बारे में. They spoke about various significant aspects. तो I went through जो Nobel Prize का officially website है, मैंने उसका पूरा content read करके, हिंदू एक्सप्रेस के सारे content read करके जो सबसे relevant चीजें हो सकती है, I've included it there. But I also went through ORF. ORF में ना, that ORF was the only website where they had spoken about way forward. What are the things that India should focus upon? I like ORF a lot because वहाँ पे ना कई बार जब detail study आएगा, they will cover a topic thoroughly, focusing on kind of कि UPSC में answers में way forward क्या होना चाहिए. ORF बहुत अच्छे से वो cover करता है. You do not have to go through it. I'll take care of it. I'll go through each and everything in ORF which is relevant here. Okay, so यहाँ पे this conclusion I've taken from there. It says that Nobel Prize for Discovery of MicroRNA signifies the importance of harnessing microRNA biology to develop new technologies and highlights the need for academic and clinical research to focus on translational application of microRNA biology. Basically बोले कि इसको Nobel Prize indicate कर रहा है कि it has a lot of potential in developing a number of new treatment and a number of new applications which can have a huge impact. It's already having a lot of impact, but its Nobel Prize indicates that it has potential to have much bigger impact on humanity. Do we get it? Do we understand it? तो फिर से एक बार सोचना कि what was this topic? This topic was about how regulation of genes happen. In our body, we have same gene in every cell, but not, we have same DNA in every cell, but in the DNA not every gene is getting expressed. Before the discovery of microRNA, our understanding what transcription factor is responsible for this regulation. Transcription factor were proteins which blocked the expression of DNA only. DNA को mRNA ही बनने नहीं देता. But then two of our scientists who got Nobel Prize this year while working on an earthworm realized that there is something called microRNA. This microRNA blocks mRNA. Gene से mRNA बन जाएगा, but mRNA will be blocked and thus gene will not be expressed. Initially when they published it, there was not a lot of significance given to it because it was considered a peculiarity of earthworm, but later they showed that microRNA exist in complex living organisms also, including humans. In humans, around 1000 microRNA have already been discovered, and thus its significance is immense in understanding regulation of gene expression in human beings, in understanding our evolution, in dealing with various diseases which may occur because of poor functioning of mRNA, and in developing future treatment also. Then it also plays an important role as a biomarker. Diagnostic maybe microRNA can play an important role. So what do we need to do? We need to develop microRNA based therapies. Already a lot of research and development is happening. That needs to be prioritized. We need to develop tools through which microRNA can be delivered. India recently biotechnology के लिए बहुत सारे initiatives लेके आया है. उसमें microRNA based initiatives have to be prioritized, and India should also focus on international collaboration in this field. We have several initiatives under which we are collaborating with different countries in the field of biotechnology. Within biotechnology, microRNA based collaboration is also something that we need to prioritize. This Nobel Prize indicates the significance that microRNA has for benefit of humankind, whether in the form of new therapies, whether in the form of overall diagnostic and improvement of human life. This is the overall summary about the topic. आपको मेंस में यही सोचना पड़ेगा, बट जब प्रीलिम्स के बारे में प्रीलिम्स में पढ़ने जाओगे ना तो थोड़ा ध्यान से अंदर की चीजें भी देखना, बिकॉज sometimes they go into, जैसे RNA interference technology, no one had ever expected that UPSC will ask question on RNA interference technology जब वो आया, राइट, बट नाउ व्हेन यू सी कि RNA interference technology पे यार नोबेल प्राइज मिला हुआ है, इतना तो पढ़ना चाहिए था. अब इन द सेम वे माइक्रो आरएनए टेक्नोलॉजी पे, अब नो वन वाज एक्सपेक्टिंग कि माइक्रो सैटेलाइट मिनी सैटेलाइट पे क्वेश्चन आएगा, दो वी वर कवरिंग दोस इन क्लासेस, बट दे आस्क्ड क्वेश्चन ऑन मिनी सैटेलाइट माइक्रो सैटेलाइट्स. अभी रिसेंटली उन्होंने क्वेश्चन पूछे थे ऐसे ही, इफ दे आस्क क्वेश्चन ऑन माइक्रो आरएनए वी शुड नॉट बी लाइक ये कुछ बहुत कॉम्प्लेक्स आ गया. एंड मोर ओवर समझो इस बात को, नोबेल प्राइज हैज बीन गिवन ऑन दिस इशू, एंड इफ वी आर एबल टू अंडरस्टैंड दैट, Nobel Prize is given on what? Nobel Prize is given on the biggest advancements which are impacting humankind. और हम वो even without even if we are not from science and technology background are able to understand, so that's a achievement for us. That shows कि हमारा overall understanding improve हो रहा है. And for those of you who may have found many of these terms very complex, don't worry. जनवरी में जब हमारा टारगेट प्रीलिम्स फेज स्टार्ट होगा, वी विल बी स्टार्टिंग विद साइंस एंड टेक्नोलॉजी, एंड देयर बिफोर जंपिंग ऑन टू दिस टॉपिक मैं वहां पे ये टॉपिक दोबारा रिवाइज करवाऊंगा आपको प्रीलिम्स के टाइम, बस प्रीलिम्स के पर्सपेक्टिव से, वे फॉरवर्ड और वो सब नहीं कराएंगे. बट वहां पे बिफोर टीचिंग दिस आई विल स्पेंड 1-2 आवर ऑन द बेसिक्स ऑफ बायोटेक्नोलॉजी. आई विल स्पेंड अ लॉट ऑफ टाइम. मैंने बोला कुछ सब्जेक्ट, साइंस, एनवायरमेंट, इंटरनेशनल बॉडीज, मैपिंग, international relations. For these things you will not be referring to any source. I'll make sure that each and everything in the most updated way is there in your notes only. आपको हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी के बेसिक्स और एथिक्स, बिकॉज मेंस का भी प्रिपरेशन कर रहे हैं, आपके टेस्ट के सिलेबस में एथिक्स भी रहेगा, है ना. विल मेक श्योर दैट बाय दिसंबर जनवरी यू आर रिवाइजिंग एथिक्स आल्सो 2-3 टाइम्स अदर देन रिवाइजिंग दीज थिंग्स. तो ये हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी के बेसिक्स, करंट अफेयर्स नहीं, करंट अफेयर्स में कराऊंगा, इकोनॉमिक सर्वे ऑफ इंडिया, बजट में कराऊंगा, मेंस के बुलेट पॉइंट का एक सिलेबस में करा के दूंगा. बेसिक्स ऑफ हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी, यू शुड नो. एथिक्स यू हैव टू डू इट योरसेल्फ. आई विल कवर सम टॉपिक्स, जैसे अब लाइफ ऑफ रतन टाटा हमें क्या क्या एथिकल वैल्यूज दे सकते हैं, क्या क्या लेसन्स दे सकते हैं, उस तरह की चीजें हम करेंगे, बट नॉट, क्योंकि इट्स मोर ऑफ अ वेरी स्टैटिक टॉपिक. हम करंट अफेयर्स में डू नॉट एम्फेसाइज अ लॉट ऑन दैट. तो आई होप ये टॉपिक माइक्रो आरएनए इज अंडरस्टूड बाय ईच वन ऑफ अस. वी आर ऑल कंफर्टेबल विद इट. ओके, नाउ विल गो टू द नेक्स्ट वन. लेट मी टेक दिस ऑनलाइन डाउट्स जरा जल्दी से. Will you cover GS2 before December like you covering topics of agriculture and disaster management GS3? नहीं भाई, I'm not promising that your current affairs of everything GS2 will be completed by this before December. Before your prelims, all your GS2 things would be completed before your mains. मैंने बोला सिलेबस का एक एक बुलेट पॉइंट कवर करना, वो प्राइमरली हम टारगेट मेंस फेज में करते हैं, जून जुलाई वाले पीरियड में, है ना. आई एम नॉट प्रॉमिसिंग इट कि आपका पूरा GS2 और पूरा GS3 कवर कब हो जाएगा, अभी दिसंबर में हो जाएगा, बट आई विल ट्राई टू फिनिश एज मेनी टॉपिक्स एज पॉसिबल सो दैट वहां पे यू हैव लेस लोड. जैसे दिस मंथ आफ्टर कवरिंग 3-4 बुकलेट्स फॉर व्हाट इज हैपनिंग इन द करंट अफेयर्स, आई विल जंप ऑन टू इंफ्रास्ट्रक्चर. मैं आपको 1-2 बुकलेट दूंगा पूरा इंफ्रास्ट्रक्चर पे, बेसिक से. मैंने आपको क्या बोला, बेसिक्स ऑफ इकोनॉमी आना चाहिए. आई डू नॉट वांट यू टू नो एग्रीकल्चर भी, इंफ्रास्ट्रक्चर भी, लैंड रिफॉर्म्स भी, जो सब सिलेबस में, वो करवाने की जिम्मेदारी हमारी है. वी विल cover that in class. Okay, now let's go to the next topic, Nobel Prize in Chemistry. सब मेरे से ही करवा लो. एक तो इसे चलने दो यार, ये बंद करवा दे यार, ये भी बंद कर दो, मेरा बंद मत करना बस, है ना. सोनू जी ये एसी बंद करवा दीजिए. सर बिग फैन फ्रॉम कश्मीर, प्लीज स्टार्ट MMP 2.0. यार MMP 2.0 लॉन्च करने का स्कोप नहीं है, समझो. मेंस मास्टर्स प्रोग्राम वो 30 सितंबर या 1 अक्टूबर को स्टार्ट हुआ है, 4 मंथ्स का कोर्स है. अक्टूबर, नवंबर, दिसंबर, जनवरी, है ना। अब मैं 2.0 लाऊंगा और फरवरी में जाएगा, फरवरी में हमारा डेडिकेटेड प्रिपरेशन प्रीलिम्स के लिए होना चाहिए। ऑलरेडी एमएमपी 1.0 इज गोइंग टिल 8th of Feb, तो रादर देन वेटिंग फॉर कि हम 2.0 लॉन्च करें, आप इसी में लो ना, पुराने वाले वीडियोस देख लो, जो अभी 2 वीक की क्लासेस हुई है वो वीडियो के फॉर्म में देख लो और आगे करते जाओ, रादर देन लॉन्चिंग अ न्यू प्रोग्राम ऑल टुगेदर। feasible है ही नहीं, it's not possible, and moreover we would not want to do that because we ourselves want to jump onto full-fledged prelims preparation from January. क्या बोल रहे हैं तरुण? तुम्हें अलग ही अपना ज्ञान देना है। चलिए, नोबेल प्राइज इन केमिस्ट्री 2024, केमिस्ट्री नोबेल प्राइज हैज बीन गिवन फॉर प्रोटीन। ये ना कौन सा, ये वाला इसी बंद कर दो, वो भी मेरा एक चलने देना। हाँ, let's come to Nobel Prize in Chemistry. It has been divided into 2 parts. इधर ध्यान देंगे। सर 10th नवंबर से ज्वाइन नहीं कर सकते, 10th को कर लेंगे। हाँ ठीक है ना, आप 10th नवंबर को ज्वाइन करिए, रोज 2-2 क्लासेस कर लीजिएगा, कुछ वैसा कर लीजिएगा। आई एम टॉकिंग अबाउट एमएमपी। किसी बच्चे को देखो, मेंस मास्टर्स प्रोग्राम इज एन एक्सीलेंट प्रोग्राम। हमने वैसा प्रोग्राम अभी तक कभी लॉन्च नहीं किया था, बट आफ्टर लॉन्चिंग इट रियलाइजिंग कि बच्चों का जीएस 1 2 3 4 का सिलेबस का एक एक बुलेट पॉइंट कवर्ड होता नहीं है अपने आप से, और यहाँ पे वी आर ट्राइंग टू इंश्योर दैट इन द 4 months। तो इट्स एन एक्सीलेंट प्रोग्राम, और जिसको भी ऐसा लगता है कि उसका सिलेबस जीएस 1 2 3 4 पूरा कवर्ड नहीं है, एक एक पॉइंट, एक एक पीवाईक्यू अगर उसने नहीं कर रखे हैं, एमएमपी विल बी वेरी यूजफुल फॉर देम, वेरी क्रूशियल फॉर देम। उसके बाद उनको बस करंट अफेयर्स अपडेट्स करने की जरूरत पड़ेगी, वो भी वी हैव अ करंट अफेयर्स प्रोग्राम, यू कैन इवन गो फॉर दिस। ओके, लेट्स टॉक अबाउट नोबेल प्राइज इन केमिस्ट्री 2024। किसको मिला है? 2 अलग ग्रुप ऑफ साइंटिस्ट को मिला है। वैसे 3 साइंटिस्ट आर देयर, बट a Nobel Prize has been divided into 2 halves. First half has gone to David Baker for computational protein design. Computational protein design means he was able to develop altogether a new protein that is not same as any of the proteins that exist. And the second half of the Nobel Prize has gone to Demis Hassabis and John M. Jumper for protein structure prediction, or for AlphaFold 2. AlphaFold 2 is a AI tool for predicting protein structure. AlphaFold 2 की प्रोटीन देखो, प्रोटीन वैसे तो अमीनो एसिड से बना हुआ है, प्रोटीन्स आर व्हाट, चेन ऑफ अमीनो एसिड, बट दे आर structured in such a manner that it has a 3D structure. तो AlphaFold 2 को आप अगर बताओगे कि which are the amino acids which are there in the protein, AlphaFold 2 will be able to predict the structure of the protein. Before this, when we had to study the structure of the protein, तो ना एकदम माइक्रोस्कोप, इलेक्ट्रॉन माइक्रोस्कोप ले लेके exactly डिटेल में जाके पढ़ना पड़ता था. We did not have a mechanism of predicting what will be the, if these are the amino acids, what will be the structure of the protein. They have developed AI tool by which they are, if you give them the which are the amino acids which are there in the protein, they'll be able to predict the structure of the protein. Do we get it? 2 लोगों को मिला है. पहले बंदे ने क्या किया, एक ऑल टुगेदर नया प्रोटीन बनाया. It has many applications. We'll briefly talk about that. दूसरे ग्रुप ने क्या किया, इट्स अ कंपनी, Google has acquired this company. It was a London based company called DeepMind. It was an AI company, and they have developed AlphaFold 2, a tool to predict the structure of the protein. Prelims के लिए एक तो पहले ये दिमाग में घुसा लो. When you go out of this classroom, you should clearly know UPSC has still not asked questions on किसको मिला है, but they've asked questions on applications and all. We should know that, and we never know क्या करने वाले. Like UPSC never used to ask last year कौन-कौन ये World Heritage Site की लिस्ट में ऐड हुआ. They never used to ask कि 2023 में कौन ऐड हुआ, but उन्होंने पिछली बार पूछ लिया, right? वो थोड़ा फैक्चुअल भी हो रहे हैं, and many of you may be preparing for UPPCS and all those exams. वहां पे ये फैक्चुअल चीजें बहुत ज्यादा रेलेवेंट हो जाती है. तो this thing is useful for your preliminary examination. थोड़ा सा समझते हैं. देखो, प्रोटीन्स generally consist of 20 different amino acids, which can be described as life's building block. Like every living organism has protein. These are the different proteins, and these different proteins, these are the different amino acids, and these different amino acids are arranged to form a protein. A protein ka 3D structure is complex. In protein, amino acids are linked together in long strings that fold up to make a 3D structure, which is decisive for protein's function. Structure plays a very important role in what the protein is doing. Since the 1970s, scientists have tried to predict protein structure from amino acid sequence, but this was notoriously difficult. Since 1970s, what has been scientists been trying to do? कि यार, this is the sequence of amino acid. देखो, जीन से क्या बनता है? mRNA. mRNA basically has details which amino acids, what sequence amino acid should be used by ribosome. So ribosome is factory. Ribosome is factory. Please understand. अभी हमने समझा था. We have DNA within the nucleus. On DNA we have genes. Gene से क्या बना? mRNA, messenger RNA. If this messenger RNA is not blocked by microRNA, है ना? अभी microRNA साइड में रख देते हैं. Forget the first Nobel Prize. This messenger RNA will go to ribosome. Ribosome is factory. क्या बनाने की? प्रोटीन बनाने की. तो राइबोसोम will get amino acids. एक और आरएनए होता है ट्रांसफर आरएनए, बट अभी उसमें जाना नहीं चाह रहा मैं. ट्रांसफर आरएनए brings amino acids to ribosome. What has messenger RNA brought? इस सीक्वेंस में इन ट्रांसफर आरएनए को अरेंज करना है. राइट. तो बाय जीन वी कैन नो कि प्रोटीन has which sequence of amino acids. Am I making myself clear? A bit. Not necessarily. Am I making myself clear? जो मैंने अभी बताया फिर से. I'm saying messenger RNA is telling ribosome कि this is the order in which you should attach protein to make protein. Which protein has to be formed? ये information कौन लेके आ रहा है? Messenger RNA लेके आ रहा है. And thus it has information about what is the sequence of amino acids. What does ribosome do? Ribosome amino acids को attach कर करके protein बना रहा है. तो messenger RNA is bringing information about the sequence of amino acids. तो by gene, from genes we can understand कि इस protein का, in this protein which are all the amino acids which are there. Can gene help us determine that? A gene can help us determine which are the amino acids that we have in this protein. But usse we are not able to determine the structure of the protein, because the 3D structure, it's a complex one, and this has been a problem that we have been facing since 1970s. Scientists have tried to predict protein structure from amino acid sequences. Amino acid sequences we know what is the sequence, but by this sequence what structure of protein would be formed, that was difficult to know. Who has done it? AlphaFold 2 has done it. A structure determines functionality. How, what role will it play? It will be determined by the structure of the protein. Okay. Did we understand the problem? Okay. Now, key achievements of the Nobel laureates. We have two different kinds of a Nobel Prize has been divided in two halves. First half was by is for David Baker. What did he do? He succeeded in using these blocks to design a new protein. He used amino. यहाँ पे blocks क्या है? Amino acids है. He used amino acids to develop a new protein that was unlike any other protein. A new protein altogether. Since then, his research group has produced one imaginative protein creation after another. That is, एक बार you develop the method of creating new protein, what can happen? You can create more and more new protein. And since then, since he has discovered the method of creating new protein from amino acid, his team has created many other new proteins, proteins which were unlike any other protein, protein which did not exist earlier. अब आप बोलोगे significance क्या है? Newer proteins may have a pharmaceutical role, may have vaccine related role, may have role in nanomaterial, may have role in tiny, basically नए protein various proteins in our body do different things. Now, when we are creating a new protein, it may have some important function, it may act as medicine, or it may be used in pharmaceuticals, it may be used in for making of vaccination, it may be used in other health purposes, and already the new proteins which have been manufactured are being used for pharmaceuticals, are being used for vaccines, are being used as nanomaterial, are being used for developing tiny sensors, etc. So, application of David Baker's work, will we be able to write it? एग्जाम में अगर मेंस में क्वेश्चन आता है कि इसके क्या एप्लीकेशन है, जो अभी रिसेंटली नोबेल प्राइज मिला है, किसमें मिला है? प्रोटीन मैन्युफैक्चरिंग में मिला है, उसके क्या एप्लीकेशंस हैं? तो प्रोटीन मैन्युफैक्चर हो गया, दिस मैन्युफैक्चर्ड प्रोटीन इज ऑल टुगेदर अ न्यू प्रोटीन व्हिच डिड नॉट एग्जिस्ट. इट मे जस्ट बी अ न्यूट्रिएंट फॉर अस, it may act as medicine, it may act as vaccination, it may be used for other things like biosensors or making tiny sensors and so on. This protein may easily get attracted to certain chemicals and thus may act as biosensor. These are all the applications. So basically नया protein बन के जो जो फायदे हो सकते हैं, we'll be making protein for all those purposes. Are we clear? Are we able to understand this? Now, Demis Hassabis and John Jumper presented an AI model called AlphaFold 2, which can predict the structure of virtually all the 200 million protein that researchers have identified. देखो, please understand, amino acid का chain है, 20 amino acids के chain है. अब how many different kind of protein can be there can be determined by the sequence. आप probability लगाओ, आप permutation and combination लगाओगे, you will see that there are many different kind of protein possible. In our body, there are genes for 20,000 proteins, but the genes that we have are just among the living organisms on earth. Our genes form a very small part, and all the genes are still not expressed. Not expressed means all the possibilities of all the possible sequences are still not expressed in nature. But amino acid से permutation and combination से 20 amino acids है, उसके permutation and combination से जितने भी क्या बन सकते हैं? Protein. Determining structure of protein was a challenge. They have done it. अब देखो, AI कंपनी, आर्टिफिशियल इंटेलिजेंस की कंपनी मोस्टली मैथमेटिक्स एंड कंप्यूटर साइंस पे काम कर रहे हैं, बट केमिस्ट्री में नोबेल ले रहे हैं. They're getting Nobel Prize in chemistry because they are helping us understand protein better, which is chemistry. Are we getting it? इससे भी बड़ा भी नीचे फिजिक्स वाला आएगा, एटलीस्ट इसमें प्रोटीन. अब देखो, पॉइंट ये है ना कि वी हैव गिवन दिस अवार्ड फॉर केमिस्ट्री, बट डीप माइंड, द सेम कंपनी ऑफ गूगल, it has also developed many other things which are being used in physics. अब for example, artificial neural network के लिए हमने क्या दिया? फिजिक्स का नोबेल प्राइज दिया इस बार. AlphaFold has been developed using artificial neural network. AlphaFold has been developed using artificial neural network. So could we not have given artificial neural network को chemistry वाला नोबेल प्राइज? Technically सोचा जाए तो that could have been given because we are using artificial intelligence everywhere, even for AlphaFold 2 development. अभी मैं इसके development के बारे में बताऊंगा, how it was developed. We'll look at overview. We'll take an overview. समझ में नहीं आता है तो ignore कर देना, because इतने डेप्थ में जाएंगे नहीं. But I wanted that वो नोबेल प्राइज वाला टॉपिक थोड़ा सा डेप्थ में ज्यादा हम कवर कर ले for better understanding. Okay, some of you have just written mains and are preparing for interviews. आपके इंटरव्यू में पूछा जाए तो आप एकदम confidently बता पाओ, especially for those of you who come from computer and IT background. You should be confidently able to answer. But come back to this. पहला, what did the scientist do? He developed new protein. He developed mechanism for manufacturing new types of protein. And since then, many different kind of proteins have been developed, which are being used for pharmaceutical purpose, which are being used as vaccinations, which are being used for nutrients, which are being used as sensors, which are being used as receptors, and so on. Then AlphaFold 2 wale scientists, they determined or predicted, determined की वजह term should be predicted. they can predict the structure of virtually all the 200 million proteins that researchers have identified, since their breakthrough AlphaFold 2 has been used by more than 2 million people from 190 countries. So applications क्या है? Researchers can now better understand antibiotic resistance, create images of enzymes that can decompose plastic. Basically ना, they're saying कि यार, once you understand the structure of the protein, you can understand its application better, and you can also understand, देखो, when a virus is impacting you, a virus is impacting you in the sense, COVID virus की बात करते हैं. COVID virus had the spike protein. It was the spike protein which got attached to your ACE receptor and entered you. Now, if you want to handle the spike protein, counter the spike protein, if you understand the structure of spike protein better, will it not be useful? Yes. तो आपके ट्रीटमेंट में भी यूजफुल हो सकता है, नई मेडिसिन बनाने में भी यूजफुल हो सकता है, नए वैक्सीनेशन बनाने में भी यूजफुल हो सकता है. Better you understand, you're able to understand these things, better it can be. Okay, examples of protein structure predicted by AlphaFold. These are some of the protein structures that have been predicted. Now, ये किस चीज की protein structure है? देखो, when you go to cell membrane, when you go to, this was our cell. Cell has cell membrane, plasma membrane. उससे material can come and go out through osmosis and all. When we'll study biology, you will see that cell membrane is made up of lipid bilayer. It is made up of lipid bilayer, and it is porous. There are proteins here which makes the cell membrane porous. तभी तो इसके अंदर न्यूट्रिएंट्स आ सकते हैं, वेस्टेज जा सकता है बाहर, है ना? तो there are proteins which are making it porous. Part of huge molecular structure in the human body, more than 1,000 protein form a pore through the membrane surrounding the cell nucleus. A cell nucleus के membrane में भी pores होते हैं. तो this has been predicted using what? AlphaFold. It was very difficult to study the structure of the protein. This they have predicted using AlphaFold. In the same way, natural enzymes that can decompose plastic. The aim is to design protein that can be used to recycle plastic. So they have seen in some living organisms enzymes which can decompose plastic. अब उसका उस enzyme का उस protein का structure कैसा है? They have predicted using AlphaFold. Once you understand the structure, it's easy to understand how it's performing. It's easy to manufacture it in future. Similarly, a bacterial enzyme that causes antibiotic resistance. The structure is important for discovering ways of preventing antibiotic resistance. This has also been predicted by AlphaFold 2. So, to fight antibiotic resistance, to develop things which can decompose what? Plastic easily, to understand your own cell and its structure, understanding the structure of protein is crucial, and AlphaFold will help us in achieving all this. Do we get it? थोड़ा साइंस ज्यादा हो गया, I'm sorry. कल आपको मैं साइक्लोन पढ़ा रहा हूँ, पहला टॉपिक. साइक्लोन में 4-5 questions have been asked in the mains examination. I have created 2-3 more questions. We should be able to answer all these 7-8 questions. जितने भी क्वेश्चन साइक्लोन के पॉसिबल होंगे, we'll study cyclone thoroughly. Then we'll आपको October 1 बुकलेट देखोगे तो उसमें in GS2 we are covering 2-3 topics. हाँ, जुडिशरी से रिलेटेड कुछ टॉपिक्स आए कि जुडिशरी में अकाउंटेबिलिटी कैसे होल्ड करते हैं, वो वाला टॉपिक, जुडिशियल अपॉइंटमेंट वाले इश्यूज रिसेंटली न्यूज में है. देन इन GS3 आई एम कवरिंग 1 or 2 टॉपिक्स. GS3 में रिसेंटली हाँ स्टेम सेल्स वगैरह पे वो थे, वो विल बी कवरिंग इट. तो टुमारो इट विल बी मोर बैलेंस्ड. टुडेज क्लास इज मोर अबाउट Nobel Prize, and that is why it's becoming a more science thing. Don't worry, in the end we'll talk about donkeys. There's a topic of donkeys. That's an interesting topic. You'll study, then you'll understand why I'm saying it's an interesting one. Okay, let's understand how AlphaFold 2 work. AlphaFold 2 काम कैसे करता है? Okay, now in order to understand how AlphaFold 2 work, we should briefly first understand what is artificial intelligence, what is machine learning, what is artificial neural network. इन तीनों चीजों को ना थोड़ा ब्रीफली समझ लेते हैं. दो जून बैच स्टूडेंट्स के लिए मैं ऑलरेडी ये डिटेल में कवर कर चुका हूं. आई थिंक आई टुक अ क्लास वेयर मोस्ट ऑफ द टॉपिक्स वर ऑन artificial intelligence, machine learning. For October batch students also, I'll in future I'll be covering it in details, don't worry about it. मैंने बोला है science environment, you do not have to worry about anything, even the static portion will be covering. But पहले, before we go on to how AlphaFold 2 functions, and specially अभी हम फिजिक्स के नोबेल प्राइज की बात करेंगे ना, वहां पे भी क्या, किसके लिए नोबेल प्राइज मिला है? आर्टिफिशियल न्यूरल नेटवर्क के लिए. So we should understand what basics of what artificial intelligence is, what machine learning is, what artificial neural network is, before being able to understand how AlphaFold 2 functions and how the last set of scientists achieve certain aspects. Now, artificial intelligence in very simple terms is like making your computer program do intelligent things, do things which are smart, which would have required humans. देखो machine क्या होता है? Machine, a car, a motor will keep rotating till the time you do not ask it to stop. It doesn't take decisions. Motor doesn't take, when you switch on the fan का motor rotate करता रहता है. But an intelligent machine will take some intelligent decision. Though this decision or this intelligence can be given, this intelligence is artificial intelligence. Machine naturally does not have intelligence. Human beings, living organisms have, machine does not. So this intelligence is called artificial intelligence. Now, you can give artificial intelligence to a computer program or a machine in multiple ways. One is you provide it some graph, in this situation do this, in this situation do this, in this situation do this, in this, some rule based system. That is one way of achieving artificial intelligence. Another is machine learning. Machine learning is a type of artificial intelligence. आप अपने कंप्यूटर प्रोग्राम को इंटेलिजेंट बना सकते हो in many different ways, by a rule based method, by a graph based method, or by machine learning. So machine learning is one way in which you can develop artificial intelligence. So what is machine learning? Machine learning learns from existing set of data. I think I always give this example. I may have given this example to you people in the past also, when I was June batch को जब मैं artificial intelligence पढ़ा रहा था. What is this example? If I have existing set of data about, let's say, lumps of cancer. I have data about lump 1, feature 1, feature 2, feature 3. ऐसे I may have 15 features of this lump, and I have this whether this lump was cancerous or it was not cancerous. Lump 2, it was not cancerous. Lump 3, it was cancerous. Lump 4, it was not cancerous. Lump 5, not cancerous. ऐसे I have information about millions of lump, and then I create a computer program which learns from it, which trains on this data. Then this model that I'll generate will be intelligent, but it has learned from existing set of data. This is machine learning. This is machine learning. It has learned from existing set of data. Now it's a learned model. अब इसे आप ना lump new दोगे. Lump new is information about new lump. It will predict whether it is cancerous or not. Now generally who does it? A doctor would have done it, but a machine is able to do it. It has developed artificial intelligence. But how did it develop this artificial intelligence? Through machine learning. Did we get it? Do we understand what is artificial intelligence? What is machine learning? Machine learning is a way in which you can achieve artificial intelligence. Then you will also hear a term called artificial neural network, ANN. Now, artificial neural network is a type of machine learning. They say artificial intelligence, you can get artificial intelligence in many different ways. One way is machine learning. ऐसे you can do machine learning in many different ways. One way is artificial neural network. What is artificial neural network? How does it function? To understand it, let me take you to this figure. So this is how our neurons function. हमारे brain cells हैं, हमारे neuron cells हैं. When they receive a signal, what happens is they pass on that signal through this synapse mechanism. And while passing on that signal, they'll realize whether this passing on in this direction was useful or not. If it was useful, they will strengthen this passage. If it's not useful, they will weaken this. This is how our brain learns. This is how our body learns. Here what we are saying is the brain's neuron cell is built from living cells, neurons with advanced internal machinery. They can send signals to each other through the synapses. When we learn things, the connection between some neurons gets stronger while others get weaker. So when we are learning something new, then some connections may become stronger and some other connections may become weaker. This is how human nervous system functions. Now, based on this nervous system, we developed computer mechanism by which computer program would learn. Existing data से आप learn करोगे. So, now let's come to, did we understand artificial intelligence machine learning? अब artificial neural network पे आते हैं. Let's come to artificial neural network. What we are saying is, artificial neural network are designed in such a way that they try to mimic how human beings learn from experience. Human being, when they are learning something, their connection is becoming, the connection between neurons are becoming strong. जब वो कुछ learn नहीं कर रहे हैं, connection between neurons are becoming weak. We'll try to create a model in such a way that when we learn something, connection gets more weight. When we do not learn something, connection gets less weight. So basically, artificial neural network or artificial neural network is structured something like this. There are these input nodes. These input nodes are, and these are output nodes. In between input nodes and output nodes, there are hidden layers. In these hidden layers, we have this kind of system. We may have multiple hidden layers. And अब देखो वो lump वाला example था, वहाँ पे we have to learn from existing data. So what would happen is, this node is connected to all these nodes. Sorry, this node is also connected to all these nodes. This node is also connected to all these nodes. All these connections are there. ऐसे ही this node is connected to all these nodes, this node is connected to all these nodes, this node is connected to all these nodes. When an input goes, ये lump है, इसका length इतना है, density इतना है, इसके अंदर these are the components, these are the constituents, है ना? आखिरी में you know कि this is cancerous. So what you will see is, it may be following some passage in the sense, let's say this is the step about what was the length of the lump. This was the step about what was the density. So when lump का input आया, length is more than 1 centimeter, more than 2 centimeter. Based on that, अच्छा length 2 centimeter तो यहाँ चले जाओ, ये 4 centimeter के लिए, 6 centimeter के लिए, ऐसे अलग अलग डेटा है. In the same way, density, length 2 centimeter, but density can be high or low. Let's say this lump that we are talking about had this density, had this feature, and was, let's say, what? Cancerous. So what, when our neural network would be learning, it will be strengthening this pathway, because this pathway is making what? Cancerous होने के chances हैं. Whereas it will be weakening other pathways. ऐसी second data will come, third data will come, fourth data will come, fifth data will come, sixth data will come, millions of data will come, and connection between nodes would be strengthened or weakened. That is, उनका weight बढ़ जाएगा या कम होगा. In the same way as neurons function. So we do not have to exactly mathematically understand how artificial neural network work. But if you are asked in the examination, what is artificial neural network? So artificial neural network is a mechanism by which we achieve machine learning. In this method, we replicate, our computer program replicate the way our brain learns. बात समझ में आई? ब्रॉडली आर्टिफिशियल इंटेलिजेंस क्या है, मशीन लर्निंग क्या है, आर्टिफिशियल न्यूरल नेटवर्क क्या है. Are we able to connect it? Are we able to understand this? Okay. Now, how did AlphaFold function? अब ये ना, even if you are not able to understand this, इसे ignore कर देना. How does AlphaFold 2 work? Chances of this being asked in the examination is very less, but AlphaFold 2 has got what? Nobel Prize. So it is the most important scientific achievement of the year. Achievement तो last 3-4 साल का है, but recognized, most important recognized achievement. तो ब्रीफली हम 3-4 लाइन तो लिख पाने के पॉसिबल होनी चाहिए. देखो, मशीन लर्निंग में फर्स्ट थिंग दैट यू नीड इज एग्जिस्टिंग सेट ऑफ डेटा, जिस पे आप लर्न करोगे. AlphaFold 2 is also a machine learned model. So what we did as part of AlphaFold 2 development, AI model has been trained on all the known amino acid sequences and determined protein structure. Already we know a lot of amino acid sequence and their structure. वो तो as input data मिल ही गया. Got it? First thing, AlphaFold 2 will have what? It will have existing data, all the amino acid protein का amino acid sequence and their structure. That is how we will, based on this learning, we will predict the structure of new protein. Please understand. अभी हमने क्या, in the older example we saw, we had millions of examples of lumps, whether they are cancerous or whether they are not cancerous. We learned on that, and then we created, then we predicted, when a new lump came, we predicted whether it's cancerous or not. Here what do we have to do? We already will learn from existing amino acid, existing protein and their structure, and then we will, when a new input will be given to us, we will be predicting whether it is, what will be its structure, what will be the, if this is the amino acid chain, what will be its structure. Got it? समझ में आया? Am I, आप लोगों की शक्ल देख के लग नहीं रहा समझ में आया. बताइए, समझ में नहीं आया? I'll explain it again. अब फिर से बता रहा हूं, इसको नोबेल प्राइज मिला है तो इतना आसान तो होगा नहीं, नहीं तो हम सब कर लेते, है ना. माइक्रो आरएनए वाला टॉपिक समझ में आया था, ये समझ में नहीं आया. आ गया तो एक्सप्रेशन थोड़े अच्छे वाले दो ना. ऐसे अबीर को समझ में आया? पक्का नहीं आया? आ गया. आशिक, इसका नाम आशिक है. हाँ सच में, आई गॉट टू नो इन द प्रीवियस क्लास, प्रीवियस जीएस क्लास, ये नाम आशिक ही रख दिया पापा ने. किसने रखा था वो नाम? फादर, मदर, बोथ, और ओनली फादर? बोथ ऑफ देम वांटेड यू टू बी आशिक. ओके, नाइस. हाँ, तो alpha, how does AlphaFold 2 function? First, you already have all the proteins whose sequence of amino acid and whose structure are known. What do we want to achieve? When we have a sequence of amino acid, what will be its structure? We want to determine that. We want to create a machine, we want to create a tool which will tell us what will be the structure of this protein if this is the amino acid. Now, यहाँ पे there were 4 steps involved. AlphaFold 2 development में there were 4 steps involved: data entry and database search, sequence analysis, AI analysis, and hypothetical structure. First, an amino acid sequence with unknown structure is fed into AlphaFold 2. With known structure तो already as input available है. An amino acid sequence with unknown structure is fed which searches database for similar amino acid sequence and protein structure. So basically, this new amino new amino acid sequence, we have put it in the AlphaFold 2 machine. Now AlphaFold 2 machine will try to see of the existing database that I have, where I know amino acid sequence and their structure also, to which does it match, or does it match exactly नहीं, to which it is similar. The AI aligns the similar amino acid sequence often from different species. You will see this is the new sequence that we have got. This is the new sequence of amino acid. We want to determine its structure, and these are the amino acids which we have from different species whose structure is already known. So AlphaFold 2 first will do what? It aligns the similar amino acid sequences often from different species and investigate which parts have been preserved during the evolution. Which parts have been preserved over the years. So this is the new amino acid. As a similar chain are existing in which of the species. Now, some things are preserved. We can see the pattern. For rest, we don't have any idea. Now we will predict the rest. How rest of them will be structured. And here they use things like amino acid which is positively charged, amino acid which is negatively charged, amino acid which is hydrophobic, amino acid which is hydrophilic. When you have to determine the structure, you will do what? The structure will be positive and negative will try to come together. Positive and negative amino acids will try to come together. Hydrophobic, hydrophilic will repel each other. Phobic will come together. एक hydrophilic होगा, एक hydrophobic होगा. They'll try to stay away. So in the amino acid sequence, hydrophilic and hydrophobic in the structure will try to make a structure where hydrophobic and hydrophilic things are coming together, where positive and negative things are coming together, and so on. In this way by multiple iteration we will determine the sequence, और वो sequence iteration के साथ हम improve भी करते रहेंगे. Okay, इतना समझते हैं, ज़्यादा और ज़्यादा डेप्थ में नहीं जाते हैं. AlphaFold 2 in this way will be predicting the structure of a protein whose amino acid sequence has been given. We have the amino acid sequence, usse we will determine the structure of the protein. Now let's come to Nobel Prize in Physics. Let's understand the Nobel Prize in Physics. Nobel Prize in Physics has been given to two scientists who contributed in the development of artificial neural network. आप बोलोगे सर ऑलमोस्ट तीनों में. फर्स्ट थिंग अबाउट वाज माइक्रो आरएनए एंड ऑल. यू यूज दैट इन द सेकंड वन केमिस्ट्री वाले में by explaining ki how protein is formed, amino acid and all. But there artificial intelligence was used. AlphaFold 2 was a machine which used artificial neural network, and here physics ka Nobel Prize is for artificial neural network. For foundational discoveries that enabled machine learning with artificial neural network. So what is artificial neural network? I think background मैंने आपको बता दिया. Artificial neural network is one way by which you achieve machine learning. Machine learning is a method by which you achieve artificial intelligence. Now this also I have explained. When the signal transfer is considered to be useful, the connection is strengthened. When it's not useful, connection is weakened. When our artificial neural network trains, it also trains in such a way that we strengthen, we give more weight to some connection and we weaken other connections. Now John J. Hopfield. John J. Hopfield did what? If you, he developed a mechanism of pattern recognition. Pattern recognition का मतलब क्या है? He said कि you give me an input pattern. You give me an input pattern and I'll predict this damaged pattern. This is the damaged pattern. This figure may have been damaged. It is closest to which of the stored pattern? It is closest to which of the pattern? Stored pattern. तो ये एक intelligent चीज है. For this you require intelligence. He developed it using a basic network which he called Hopfield network. In Hopfield network, some preliminary artificial neural network wala methods were used, where similarities are strengthened and then database se comparison ho raha tha. The later scientist, the second scientist, what he did, he worked on this model. He worked on Hopfield network and developed a new method called Boltzmann machine. This Boltzmann machine was able to develop a more complex system. The Hopfield network was a simpler system. he was able to develop more complex system where there can be multiple layer. One layer is connected to only the second hidden layer, second layer is connected to third layer. ऐसे one layer need not be connected to each other. So the first method, the first model, every node can be connected to every other node. In the second method, the more advanced method, what they did, one layer will be connected to next layer only. This kind of, in this way, improvements were made, and eventually what was developed? Artificial neural network was developed. And artificial neural network we are using everywhere. जहां पे भी हम artificial intelligence nowadays use कर रहे हैं, be it in AlphaFold, be it in image recognition, be it in generative AI, हमने बताया था generative AI, it is using artificial neural network, be it in deep fakes, when we are making deep fakes, that is using artificial neural network, audio generation, that is there also we are using artificial neural network. So artificial neural network is used everywhere. Here we have some examples of physics where artificial neural networks are used. For example, they are used in data analytics modeling, which is used in physics, but more importantly in medical science. X-ray images and other images from the field of medical science are being analyzed by AI to identify tumors. देखो neural network में first thing what did he do? Hopfield network, image recognition ही किया था, pattern recognition. Now X-rays का pattern recognition कौन कर रहे हैं? AI tools के through हम कर रहे हैं. Similarly in space sector, when we have images of far away universe, वहां से this is galaxy, this is star, these things of a very far image can be identified using what? Using artificial neural network. AI can be used to determine the trajectory of asteroid, to predict the trajectory of asteroid, to understand if the asteroid will be a threat to humans or not. So all these are applications of artificial intelligence in space, where we are using artificial neural network. So this is the argument which the committee is giving, why artificial neural network can get Nobel Prize in physics. It can get Nobel Prize in physics because artificial neural network is being used in all these areas of physics. Other than modeling and all, some other direct applications are also there. Now negatives are also there. Negatives क्या है कि यार machine learning is mathematics and computation and not physics. You are discouraging physicists here. यहाँ पे शेल्डन कूपर को बोल दिया जाता कि इंजीनियर्स को मिल रहा है फिजिक्स में नोबेल प्राइज, वो बेचारा सुसाइड कर लेता. इफ यू वॉच्ड बिग बैंग थ्योरी, है ना, इफ हार्वर्ड वुड हैव गॉट नोबेल प्राइज, it would have been less painful, but what was that person? हाँ, वॉलोविट्स को मिल जाता तो, और वॉलोविट्स वाज व्हाट, वो इंजीनियर था, वो कंप्यूटर प्रोग्राम लिखता था, वो सब करता था. यहाँ पे उनको फिजिक्स में नोबेल प्राइज मिला है. सेकंड, Nobel committee is very behind in recognizing and awarding contribution of ANN. ANN was developed way back. आप chemistry में AlphaFold 2 which was developed in 2020-21, you are giving Nobel Prize to them right now. ANN and its impact has been huge already. If you wanted to give Nobel Prize on that, it could have been given earlier only because it has already had a lot of impact on human beings. Then Geoffrey Hinton himself has highlighted the threats that AI pose for humankind. It says that the way AI is being developed and the way AI is gaining intelligence, it may very quickly take away our jobs, make a lot of us unemployed, but वो short term challenges है. But he's saying कि it may also become a threat to humankind because artificial neural network in this way may get more and more intelligent, and a time may come where they are more intelligent than human beings. This may become a cause of concern. जिनको भी Nobel Prize मिला है, he is considered the godfather of artificial intelligence, and he is raising these concerns. And other experts like Elon Musk has also said that one of the biggest threats that humankind face is from the field of artificial intelligence. Okay, so this was Nobel Prize in what? Nobel Prize in physics. Broadly, do we have clarity on Nobel Prize in biology, Nobel Prize in chemistry, Nobel Prize in physics? Now, I think ये तो हमने already कर लिया Nobel Prizes वाला. Let's do the last topic. अच्छा, there were some places in news. अभी ना बाकू में क्या होने वाला है, बाकू में COP29 of United Nations Framework Convention on Climate Change has to be held. तो प्रिलिमिनरी एग्जामिनेशन में दे गिव यू मैच द फॉलोइंग के फॉर्म में, ये जगह कौन सी कंट्री में लोकेटेड है, कौन से कॉन्टिनेंट में लोकेटेड है. फॉर दैट बाकू वाज इन न्यूज रिसेंटली. इट इज द कैपिटल ऑफ Azerbaijan and is the largest city on the Caspian Sea. You will see this is Caspian Sea. On Caspian Sea, the largest city is Baku. Not only this, this is a city which is located 28 meters below sea level. Caspian Sea itself is at very low depth when compared to sea level. Sea level, by here sea level we meant ocean का जो level होता है. Caspian Sea already is a lot of depth. Baku is located 28 meters below sea level. अगर आप इंडियन ओशन में जाके लेवल ऑफ वाटर देखोगे एंड बाकू का लोकेशन देखोगे, बाकू इज लोकेटेड 28 meters below that and is the lowest lying capital in the world and the largest city in the world below sea level. इससे बड़ा कोई और शहर नहीं है जो below sea level है and it is also the lowest lying capital in the world. So Baku has this significance also, this important aspect also. Now बाकू इज लोकेटेड ऑन कैस्पियन सी. अभी ना डाउन टू अर्थ में ये आर्टिकल था कि कैस्पियन सी जहां पे यूनाइटेड नेशन फ्रेमवर्क कन्वेंशन ऑफ क्लाइमेट चेंज का सीओपी होने वाला है, itself is facing a lot of challenges because of climate change. So when countries meet here, they should be focused on dealing with its challenge also. So Caspian Sea, if you look at Caspian Sea, on this side we have Central Asia, on this side we have Europe. इस तरफ क्या है? वेस्ट एशिया. ईरान को सेंट्रल एशिया में मान लो, इधर से ये पूरा वेस्ट एशिया. कैस्पियन सी इज लोकेटेड हियर. नाउ एक चीज जो प्रिलिमिनरी एग्जामिनेशन में आएगा कि विच आर द कंट्रीज विच आर सराउंडिंग कैस्पियन सी. तो यहां पे दीज आर द कंट्रीज. जैसे लोग इसे याद रखते हैं तारीख के फॉर्म में, TARIK, बट आई डोंट प्रेफर दैट. मुझे ना ऑर्डर भी बहुत जरूरी है, लाइक विच इज द ऑर्डर ऑफ द कंट्री. And इसलिए ना I use the short form of KTR. Kazakhstan, Turkmenistan, Iran, Azerbaijan and Russia. Kazakhstan, Turkmenistan, Iran, Azerbaijan and Russia. Azerbaijan का capital Baku, which is located on Caspian Sea, is the lowest city, lowest capital to be located there. Where what is going to be held? United Nations Framework Convention on Climate Change का COP29 is going to be held. So one thing is कैस्पियन सी की नेबरिंग कंट्रीज क्या है, तारीख के फॉर्म में याद रख सकते हो, बट फॉर मी सीक्वेंस बिकम्स वेरी इंपॉर्टेंट. इट मेरे को ना रिकॉल करने में बहुत इजी कर देता है, बिकॉज कजाकिस्तान, तुर्कमेनिस्तान, ईरान, अजरबैजान, रशिया, कटियार में द तारीख में वो ऑर्डर थोड़ा सा गड़बड़ होता है. नाउ व्हाई कैस्पियन सी इज ड्राइंग अप. तो देखो कैस्पियन सी रिसीव्स वाटर फ्रॉम मेनी डिफरेंट रिवर्स, बट 80% ऑफ इट वाटर कम्स फ्रॉम वोल्गा रिवर. And Volga river पे over the years a lot of dams etc. have been created which has reduced the amount of water reaching Caspian. तो Caspian तो वैसे ही छोटा हो गया when compared to previous period. But even in recent times, because of climate change, several issues are occurring. Climate change is leading to rainfall deficiency in the region. अगर rainfall deficiency होगा तो would in rivers less water would come and less water would reach Caspian Sea. तो it's impacting the precipitation in the region. तो Volga river accounts for around 80% of the water brought into the Caspian Sea. उस पे कोई challenge होना issue होता है. तो would drying of Caspian Sea impact India? It may impact Eurasia region drastically, but it will also have a lot of negative impact on India. Down to Earth had also spoken about this. It said that western disturbance originates also on Caspian. Mediterranean Sea से originate होता है, but Caspian Sea also plays an important role in the origin of western disturbances. And अगर Caspian Sea dry हो गया, it may also impact western disturbance in India, the rainfall in India, creating issues, creating challenges, and thus we should be worried about this also. Okay, तो Caspian Sea के बारे में, first location, the countries which are surrounding it, biggest city on Caspian Sea, Baku, which is also the lowest city. Baku किसका capital है? अजरबैजान का. एंड नेबरिंग कंट्रीज अगर कटियार से याद रखोगे इट विल बी इजी, कजाकिस्तान, तुर्कमेनिस्तान, ईरान, अजरबैजान, रशिया. तारीख से भी याद रख सकते हो, पर तारीख में ना ऑर्डर थोड़ा मिस हो जाता है, गड़बड़ हो जाती है. इसलिए I want you people to remember this order also. You will see that when February March में we will cover all the mapping sites. I told you कि for mapping you do not have to do anything. At that time when we are doing things in sequence, it's very easy to remember, recall. When we are not doing it in sequence, it gets confusing. एक बॉडी के अराउंड आप ट्रिक बना के कोई, बट अगर आप एक बॉडी के अराउंड चीजें सीक्वेंस में पढ़ोगे इट विल बी मच इजियर. ओके, लेट्स नाउ टॉक अबाउट डोंकी. देखो अभी ना अ नंबर ऑफ डोंकीज वर इन न्यूज. विल टॉक अबाउट ईच वन ऑफ देम जैसे जैसे वो न्यूज में आ रहे थे. बट गुजरात में एक डोंकी स्पीसीज है, नॉट द इंडियन वाइल्ड एस. इंडियन वाइल्ड एस इज नॉट इस स्पीसीज से इन एनी वे कनेक्टेड. तो इट वाज इन न्यूज बिकॉज ऑफ स्वीटनेस ऑफ इट्स मिल्क एंड पॉपुलेशन बीइंग वेरी स्मॉल. जो डोंकी या गधा जिससे हम काम करवाते हैं, इट इज नॉट related to Indian wild ass. It is related to African wild ass और African donkey. तो जो गधा काम करता है, जैसे आप राजस्थान में जाओगे या फिर दूसरे ट्रैक्स वगैरह पे जाते हो तो वहां पे बहुत सारे गधे यूज हो रहे होते हैं. दे आर डोमेस्टिकेटेड वर्जन ऑफ African wild ass and not Indian wild ass. Note it, first thing. Some people say that they have become a separate species altogether, and that's why there are two scientific names which we'll use. But first, let us start with African wild ass. What is African wild ass? It's a animal species. It's a mammal which is found in northeastern Africa. Northeastern Africa में Sudan, Ethiopia, Djibouti, वहाँ पे wild में. Please understand, we're not talking about the domesticated version. We're talking about the wild species. Now, these donkeys prefer desert environment with less rainfall. These are mountainous region where individuals can be found from sea level to 2000 meters. That is, donkeys, ये wild donkeys कहाँ मिल रहे हैं? In Sudan, Ethiopia, Djibouti, they are found from desert or zero height from sea level to around 2000 meters. बहुत cold conditions में you will not find them, and they breed only once in a year between March to September. So they have a breeding period. Preliminary examination में this kind of question has also been asked in recent years. The lion has a breeding season or not? Lion का नहीं होता, lion कभी भी mate कर लेंगे, but they have a breeding season and they are critically endangered. And why I specifically covered it? Because the species is critically endangered. Its population is going down. Sir, for science and tech and environment, no need for foundation classes. यार फाउंडेशन क्लासेस को भी मैं करंट अफेयर्स में ही पढ़ाता हूँ, साइंस और एनवायरमेंट, है ना. तो डोंट वरी, जितना करंट अफेयर्स में साइंस और एनवायरमेंट हम पढ़ाएंगे, जनवरी में मैं साइंस शुरू करूंगा. आई विल टेक अराउंड 15-20 क्लासेस फॉर इट और 50 आवर्स ऑफ क्लासेस फॉर इट. यू विल नॉट नीड टू डू एनीथिंग बियॉन्ड दैट. ऐसे ही अराउंड 20 जनवरी से 15 फेब तक मैं एनवायरमेंट कराऊंगा. यू विल नॉट नीड टू डू एनीथिंग बियॉन्ड दैट. ओके, डोंट वरी. लेट्स कम बैक इसके बारे में बात करते हैं. तो African wild ass, what is IUCN status? Critically endangered. Where is it found? Northeastern Africa desert region में. Now इसी का domesticated जो version है, that is, देखो इसका scientific name हमने क्या बोला था? Equus africanus. इसका domesticated version है Equus africanus asinus. But some people consider that जो domesticated गधा है, that is a different species, and उसको ना फिर वो दूसरा नाम देते Equus asinus rather than Equus africanus asinus, which would have been subspecies. Equus africanus is the wild animal. उसका subspecies बनाना होगा तो Equus africanus asinus. But some people consider it, some experts consider it to be a separate species. Domesticated version has become a separate species altogether and give it a different name, Equus asinus, by groups which consider it a separate species. ठीक है. Now conservation status, domesticated version का not evaluated. They are used as drought and pack animal. Update, अभी recently ना, it was in news, donkey was in news because बेटा पैक एनिमल ना, आप जब उस पे सामान लाद के जा रहे हो, ड्रॉट एनिमल जो दूध नहीं देते उनसे बाकी काम करवाते हो ना, दोज आर ड्रॉट एनिमल. नाउ कमिंग बैक टू, अभी इट वाज इन न्यूज. व्हाई डाउन टू अर्थ स्पेसिफिकली टॉक्ड अबाउट donkey was बिग बॉस. यू फॉलो बिग बॉस? नहीं, चलो अच्छा है. कोई फॉलो तो नहीं करता? कि उसमें गधे भी आने लगे. पहले भी आते थे, अब तो. दिनेश तुम फॉलो करते हो ना? सच बताना, सच बताना. तो तुम्हें कैसे पता उसमें गधे? तो ये उसे गया गुजरा. तो इस बार ना उसमें एक गधा है. बिग बॉस के घर में एक गधा है. नहीं, लिटरली गधे तो हमेशा ही होते हैं, बट इस बार थोड़ा समझदार वाला, जैसे जबरदस्ती वहां ज्यादा बड़े गधे कौन है? तो जानबूझ के वहां रह रहे हैं, है ना, ये वहां रहना नहीं चाहता है, इस डोमेस्टिकेटेड डोंकी को भी वहां रख दिया है उस हाउस में। तो डाउन टू अर्थ में एक आर्टिकल था कि दिस इज रॉन्ग बिकॉज दे आर प्रे एनिमल्स, so they are naturally nervous and in light and all these conditions it's not good. More importantly, they were talking about ki we have degraded donkeys as stupid animal. They are not stupid, they are quite intelligent. हमने वो वैसा परसेप्शन बना दिया, इस वजह से ना दोज एनिमल्स आर नॉट रिस्पेक्टेड, आर नॉट ट्रीटेड, तो उस वजह से देयर वाज आर्टिकल अबाउट डोंकी। तो आई थॉट कि डोंकी का बायोडाइवर्सिटी पर्सपेक्टिव से वी शुड नो थिंग्स, बट अ फ्यू डेज बैक देयर वर न्यूज अबाउट सेवरल ब्रीड्स आल्सो। देखो, डोमेस्टिकेटेड डोंकी is what? Domesticated donkey is the African wild donkey का एक subspecies, right? Now this domesticated donkey in different geographical region have developed different breeds. For example, in Spiti region is Himachal Pradesh वाला region, बहुत ठंडा वाला region, we have Spiti donkey. In Gujarat we have Halari donkey, Kakshi donkey. 3-4 days back Halari donkey was in news. Halari donkey is quite expensive, 1-1 लाख रुपए में एक donkey आता है. Its milk is very sweet and is in demand, and they are very docile in temperament and are used as pack animals during pastoralist migration and for transportation in the donkey cart. Their population is also very less, around 500, and thus are endangered. This breed is endangered. Entire donkey population is not evaluated. Wild donkey is critically endangered. Domesticated donkeys are not evaluated, but this one, Halari donkey of Gujarat, is endangered. ऐसे ही there is Kakshi donkey from the Kaksh district of Gujarat. They are primarily used for agriculture purpose like intercultivation for weed removing and so on. Got it? So these are different breeds of donkeys: Piti, Halari, Kakshi. Now a different species altogether is Indian wild ass, Equus hemionus khur. This is a different species. This is not African wild donkey. This is Indian wild ass. Now this Indian wild ass, you will see, or Asiatic wild ass, are also known as onager, and Indian wild ass is one of the 5 subspecies. So this Asiatic wild ass has 5 subspecies. One of it is what? Indian wild ass. We are talking about Indian wild ass. We are not, African wild ass is different, its subspecies are different. This is not related to it. गुजरात में इंडियन वाइल्ड एस मिलता है, गुजरात रन ऑफ कच्छ में. इट इज नॉट रिलेटेड टू अफ्रीकन वाइल्ड एस. दो सेम जीनस है, वो भी इक्वस है, ये भी इक्वस है, बट it is a subspecies of Asiatic wild ass. First thing. Then in the past it was very well distributed. It was distributed from Iran to West India. तो राजस्थान में, गुजरात में, महाराष्ट्र में, पाकिस्तान में, अफगानिस्तान में, ईरान में, it was found everywhere, but today its population is only confined to Gujarat of India. It is the only place in the world where it is found. Its population has been going up and thus it is near threatened. पहले ये 2016 तक it was endangered. What are the threats that it is facing? One is habitat degradation. They are losing their pastoral land. Invasion of Proposis zuliflora. Proposis zuliflora is invasive species. ये कांटों वाला plant होता है and it takes up all the resources and does not allow other grass and other plants to grow. Then encroachment and grazing by Maldhari tribes. Maldhari tribes, their animals graze a lot and do not leave a lot of food for the wild animals. And human wildlife conflict has also become an issue because 1/3 of the population of Indian wild ass lives outside protected area. Outside protected area humans would also be there, and human wildlife conflict is becoming a issue. Abhi recently Indian wild ass was in news because its population estimation happens every 5 years. Its population estimation was held census 2024 by Gujarat Forest Department. What is it? What does it say? Population has increased by 26% to 7672. Its population has increased to 7672. It inhabits 6 districts of Gujarat: Surendranagar, Kutch, Patan, Banskantha, मोरबी एंड अहमदाबाद. यहाँ पे ना आई डोंट नो वो इमेज कहाँ गई, मैंने, हाँ ये रहा ना. इफ यू विल कम हियर, कच्छ, पतन, पाटन, पाटन, एंड दिस इज बनसकंठा. Banaskantha. These 3 districts and Morbi, Surendranagar, Ahmedabad. These 3 districts. These are the 6 districts where what is found? Indian wild ass is found. Near threatened hai, but population in recent years has been going up. Okay, so this was it about Indian wild ass, but important topic है for preliminary examination. Indian wild ass न्यूज में भी रहता है और इसके प्रोटेक्शन पे भी बहुत सारा काम हुआ है, इसका सेंसस भी हुआ है. From that perspective, the question may be asked, or you should know how Indian wild ass is different from African wild ass. Indian wild ass is a subspecies of Asiatic wild ass and not African wild ass. Whereas जो बाकी domesticated animal है, which donkey has been domesticated? African wild ass. It has been domesticated. The rest of the donkeys that you see जो domesticated है, they are the African version. Okay, so this was booklet 1. आज हमने थोड़े factual चीजों पे ज्यादा ध्यान दे दिया है. Tomorrow we will be completing doing October part 1. आज हमने October part 2 किया है. Tomorrow we'll be doing October part 1. October part 1 में there is cyclone वाला topic, judiciary से related topic and some other stem cells and associated issues वाले topic. We'll do that. Okay, चलिए. Thank you very much. Any doubts? बेटा जंगल में जानवर है, वो अपने इकोसिस्टम में रोल प्ले कर रहा है ना. हाँ, डोमेस्टिकेटेड नहीं है. इंडियन वाइल्ड एस इज नॉट डोमेस्टिकेटेड एंड ए हर्बीवोरस एनिमल विल प्ले इट्स ओन रोल इन फॉरेस्ट. आप सारे हर्बीवोरस एनिमल को हटा दो तो ग्रासेस इतने बढ़ जाएंगे, है ना. यू विल नॉट हैव दिस थिंग कार्निवोरस एनिमल हु आर सर्वाइविंग ऑन हर्बीवोरस एनिमल. वैसे जंगल के हर एक जानवर का ऐसे अलग अलग यूज नहीं है ना. ऑल ऑफ देम आर प्लेइंग देयर इकोसिस्टम रोल. ओके, चलिए. थैंक यू वेरी मच. Tans is asking to what extent specifics of Nobel Prize be remembered. प्रिलिमिनरी एग्जामिनेशन के लिए ना बस समझ के रखो ना. मेंस में बाकी चीजें चाहिए होंगी, बट प्रिलिमिनरी एग्जामिनेशन में से थोड़ा समझ के रखो. बाकी बहुत ज्यादा फैक्चुअल या टेक्निकल डिटेल